in a few seconds, and I'll let you know, Anupama, and you can get going once that's done. I guess we started. Yeah, go for it.
Okay. Hello, everyone. Welcome to the third day of the NeuroFemIndia 2021 conference organized by BiosWatch India. This conference is being run in collaboration with the ALBA network, which has generously agreed to sponsor the event, and our media partner INSTEM. So my name is Anupama Satyamurthy. I am a DBT Ramalingaswamy Fellow at the Indian Institute of Science, Bangalore. I'll be moderating today's sessions with Poonam Thakur, who is an assistant professor at ISR Trivandrum. So both of us are members of the core organizing committee for this conference. Volunteers Arushi Batra, who is a PhD student at CSIR IGIB Delhi, and Tasneem Mustafa, who is an alumna of ISR Pune, will be helping us with the technical requirements of Zoom and ensuring that we can have a productive conversation with our speakers and all of you, our audience. So today we're going to have talks by 5 speakers instead of 6. Unfortunately, one of our speakers, Dr. Jayalakshmi Krishnan, won't be able to join us. And these talks will be followed by private breakout rooms from 4 o'clock to 4:30 p.m. Indian Standard Time for extended discussions between the speakers and selected members of the audience. Since we wanted to maintain a small number of participants to enable meaningful breakout room interactions, we prioritized trainees and students to participate in these. Now, in the spirit of building community, though, we encourage all participants to reach out to the speakers for any additional questions. And before we jump in, I just wanted to start the day off with 3 general notices for the audience. To foster a spirit of inclusivity and openness through this conference, we kindly request our audience members to think about adding their preferred pronouns to their Zoom names in solidarity with people of underrepresented genders. You can do this by adding preferred pronouns and brackets after your name in your Zoom profile. Second, at this point, we would like to remind you that we are also live streaming this conference on YouTube on BiosWatch India's channel for any members of the public and the Indian academia who missed out on registering for the event. So the link should be displayed in the chat shortly. We'll be monitoring the YouTube chat stream also, in addition to the one on Zoom, to select audience questions for our speakers. Third, we'll not be accepting any audience questions for the field overview talk alone, but for all the other talks and sessions, we highly encourage the audience to ask their questions in the Q&A pop-up box. So please upload the questions that you want the speakers and the panelists to answer, and please address your questions to specific panelists by name. You're welcome to use the chat box for public discussions, but for the questions, it's much better to add questions to the speakers in the Q&A pop-up box. So that they can be upvoted and all volunteers and MCs can find them easily. With that said, let's kick off the talks for the day. So please join me in welcoming Dr. Shubha Tole, who's a professor in the Department of Biological Sciences, TIFR, Bombay. In her research, Dr. Tole investigates the genetic and epigenetic mechanisms that control the formation and development of the cerebral cortex and the amygdala in the brain. She's won the 2010 Shanti Swaroop Bhatnagar Award in Biological Sciences and the 2014 Infosys Prize in Life Sciences. She serves as the chairperson of the Women in Science Committee of the Indian Academy of Sciences and is a member of the ALBA Network Steering Committee. So today, Dr. Tole is representing the subfield of neuronal development and will give a talk focused on the history and importance of this field. Dr. Tole, thank you for accepting our invitation to talk in this conference. The floor is all yours.
Thank you so very much. Am I audible?
Yes.
So it's a really special opportunity. I think of all the conferences I've spoken at, I think this is by far the most unusual and the most special. And I'm really, really delighted to be here today. And particularly delighted to be sort of introducing the field of developmental neuroscience, a field close to my heart. Oh, sorry, some random chat has come in the middle. Now, why is developmental neuroscience special and what is unique about it? Oftentimes students come to me very interested in the brain. Oh, why the brain? Well, of course, we're all neuroscientists. We all came and sucked in by, color vision or dreams or learning and memory or mood or emotion. We all came in because of some aspect of brain function that we found particularly intriguing. Now, so my lure was vision. I wanted to, visual perception was my sort of thing. And when I began graduate school, I realized that everything the brain does is a result of circuitry that is wired up properly. And if the circuitry does not assemble properly, the brain function is not possible. So in some sense, how the circuits grow enable the brain functions we have, and they also constrain them. There are certain brain functions we cannot generate because we just don't have the wiring for it. It's sort of like asking, it's like asking a toaster to, run a software program or something. The wiring is not there, you can't do it. So development in some sense underlies all function, absolutely all function. And that is the lure of development, because you're studying the assembly of the computer. It's sort of like a self-assembly system where, you have to 3D print every part, as in develop it, self-ate specify from progenitor cells, and then you have to assemble it all together. So that's what makes developmental neuro really special. Now, special as it is, this field actually has an entire sort of theme devoted to it in the SFN themes. They have theme A, B, C, D, E, and theme A is development. Okay, so it's sort of a giant chunk of all of the various types of subcategories of neuroscience. And yet in India, this is an extremely poorly represented field. Okay, now neuroscience itself in India has not been that well represented until recently, and I'm delighted to see so many neuroscience, young neuroscientists, and especially women neuroscientists in India in this meeting today. But broadly, neuroscience itself is not well represented, and there's a reason. In the undergrad curricula all over India, biochemistry and genetics are extremely well taught, immunology. So these are the things that our young students get enamored by, because any intelligent mind is going to get attracted by what they get exposed to. And then we, go off to do further studies and PhDs in these fields, and which is why we are very, very rich in these fields that are taught very well at undergrad. So in some sense, the solution is to start introducing basic neuro courses at undergrad institutions. And I'm saying this in this forum because there are many, many young people here who can motivate this process at their institutions. Developmental neuroscience, well, even more a niche field. So I allowed myself this sort of longish introduction because the history of developmental neuroscience in India is very, very short. It's very, very small. And so you heard some of this perhaps in Vidita's talk a couple of days ago. Neuroscience in India started from the neurology, the neuropsychiatry angle, medical trained people contributing to neuro. And indeed, some of the early development type of studies were essentially child development, imaging children's brains in many different ways and looking at it. Now, what we regard as modern mechanistic development, genetic mechanisms underlying development.
Video name.
So what we regard as modern mechanisms that underlie development, that work, I would say, was pioneered in India by an amazing neuroscientist whom I was proud to have as a colleague and indeed chairperson of my department, Veronica Rodriguez. So I'm going to start showcasing some of her work. So this is Veronica. She's no more. She passed away a few years ago, a great loss. And some beautiful articles have been written eulogizing her, which I will post on the chat afterwards. But I would like to showcase her contributions to developmental neuroscience. Veronica did her PhD with Obaid Siddiqui at TIFR. And Obaid's interest was the neurogenetics of olfaction. how olfactory behavior is learned. And so it was the behavior and the function of olfactory. Veronica did a short postdoc in which she looked at olfactory glomeruli and in fact did mapping of how olfactory glomeruli are sort of specific to process different odors. But when she returned to TIFR as a faculty member, her interests began taking on the sort of flavor of, well, how does the circuit get assembled? She created this field in India, which was then a very niche field, as to how does the circuit get created that underlies all of these behaviors that people have been studying, olfactory behaviors in the fly and so on. So I'm going to just show a few titles of papers that she published and then showcase one. Okay, now this is some of her early work, and you can see this is Krishanu Ray, who is currently the chairperson of our department in TIFR, so it's a nice full circle. Cellular events during development of the olfactory sense organs in Drosophila. And then another in the sort of same general space, development of the Drosophila sense organs, utilize cell-cell interactions as well as lineage. So sort of very cellular genetic mechanisms of development. And then this beautiful work, Dhanisha Javeri, who is now a senior scientist at Queensland Brain Institute in Australia, in Brisbane. She was a PhD student when, in fact, I was a very young faculty at TIFR. Sensory organs of the atonal lineage, pioneer of the formation of glomeruli within the adult Drosophila olfactory lobe. Now, this study was interesting because atonal is a BHLH transcription factor. And these are usually, these BHLH factors are thought to control cell lineage. Much work has been done in that space. And here they're showing that, well, they also control circuitry because the glomerulus is essentially neuropil, as the fly people would call it, white matter, as the mammalian people would call it. And that this atonal lineage pioneers the formation of glomeruli is what this study showed. Now, other sort of studies in the same general space of glomerular and olfactory lobe patterning. Again, Dhanisha Javeri, positioning sensory terminals in the olfactory lobe of Drosophila by robo-signaling this time. So then Veronica's work moved to signaling molecules, and they've been, I'm not going to, this is not her whole CV, okay? These are just highlights which I've selected for some reason. But work from her lab has looked at robo-signaling, efferent signaling, Wnt signaling, in circuitry. So this assembly of circuitry is something that Veronica's lab produced rich findings in. And I'm going to sort of showcase who she was as a scientist with one story, because I cannot do justice to everything that Veronica was and how she led not only our department, but our institute's biology efforts, and indeed impacted the entire country and people outside. But I'm going to showcase who she was as a scientist with this one story. And it's this, okay? Neuronal activity and Wnt signaling act through GSK3 beta to regulate axonal integrity in mature Drosophila olfactory sense organs. Okay, now before I go to the next slide, which showcases this, I'm going to sort of point out that look at just, this is a selected publication history, right? And look at how every few years there are publications in this, very, very top line, fantastic, a journal of fantastic studies, development. And this sort of was something that inspired me. So I joined TIFR in '99, and I took a look at what Veronica had achieved in, the decades before. And I figured, wow, if a scientist like Veronica publishes this regularly in a journal which I regard very, very well, then if I'm not able to do the same thing, then the problem falters with me. So, this is doable, science in India can flourish, was sort of something that inspired me. Okay. Okay, so I'm going to, in fact, focus on this study, dendritic refinement of an identified neuron in the Drosophila CNS is regulated by neuronal activity and Wnt signaling. Okay, so this is the study I'll be showcasing. And I'm reproducing the title here. Okay, what does this study show? And why does it, why have I selected this? I've selected this study because of this quote from Krishanu Ray, who was Veronica's PhD student and is now the chair of our department. He said, Veronica was always curious to probe uncharted territories, and this paper is an example of a fearless mind. Also, she was prepared to persist to conclude whatever she started. This project started as a simple curiosity to understand how a dendritic branch, an important contemporary question in the neuronal circuit development, how a dendritic branch is produced. And then an MSc student accidentally found a way to selectively label these particular neurons. So Veronica picked this most unusual neuron to probe this question, and she brought it to a conclusion. So what did she find? I'm going to show you one panel from this paper. Okay, so this row on the left is control. Okay, and here is when activity in this neuron is inhibited. And you can see through development, larval stage 3, the two look similar, and then puparium formation stages. And you can see what happens in the control is that these dendrites which branch get curtailed and pruned. And then this is, sort of one major solid branch. And look what happens here. Okay, these extraneous branches do not get pruned. Okay, and this beautiful arbor is because this neuron has been curtailed in its ability to process activity. And this, in the paper, they link to a few very, very important molecules. So I've just excerpted bits of the abstract just to showcase how creative this study is. The abstract reads, paraphrased, here we study a central serotonergic neuron in the Drosophila brain, the dendrites of which undergo dramatic morphological change during metamorphosis. Sensory input, possibly acting through NMDA receptors, is necessary for dendritic refinement. Our results indicate that activity triggers Wnt signaling, which plays a pro-retraction role in sculpting the dendritic field. Okay, in the absence of sensory input, dendritic arbors do not retract. And this phenotype can be rescued by activating Wnt signaling. So see how she's connected activity and NMDA receptors and sensory input to Wnt signaling. Our findings indicate sensory activity, NMDA receptors, and Wingless/Wnt5 signaling pathways to advance our understanding of how dendritic refinement is established. Okay, and here is just, this picture will stay with me forever. So this sort of showcases a unique scientific mind, and I can't do justice to everything that Veronica was and how much of an impact she had on all of us. So I will post links to a beautiful sort of write-up about her after I finish this presentation. But so Veronica, in some sense, is the first developmental neuroscientist in India. And happens to be a woman, but there weren't very many others. I mean, occasional collaborators of hers have done a few things, but she was essentially the person who started it. Inspired by her, younger people joined. So I'm not going to talk about my science, but I'm going to talk about my story a little bit. I joined full of idealism in '99, thinking, oh, well, developmental neuroscience, I have a fantastic role model, and TIFR really wanted to start vertebrate developmental neuroscience. So my lab was the first vertebrate developmental neuroscience lab in India. And I was boldly, venturing in saying, okay, and I joined TIFR, which essentially didn't have an animal house. Okay, so all those plans came to a screeching halt. What it had was a mouse holding facility in which a colleague, Shobna Sharma, would bring in mice and infect them with malarial parasite and then study them. But they didn't actually have a mouse breeding facility. So in fact, for my entire first year of my lab, I would fly in mice, timed pregnant mice from the University of Hyderabad. And then we would have, the mice got placed near a very noisy propeller. And so they, aborted because the noise was too much for them. Or, the hold of the particular flight that we're putting them on isn't pressurized, so we can't send the mice. And all of this happened. So there was mouse issues and there was personnel issues. So what personnel issues? Okay. I found, I'm just going to stop sharing the screen for a minute. I found that because neuroscience is not well represented, was not well represented in India and was not taught at undergrad, I found that the best and the brightest didn't know about it. And they were unwilling to take the risk of learning something new. So in fact, when I joined my very first year, 99, the group of first years, went around meeting all the faculty. The, sort of bright star of that group, now a successful faculty member, sat in my office and very honestly said, Shubha, I find your work interesting and cool, and it's something very different that I've never heard of. But what scope is there in this field for the future? He really wanted to know what scope there was in this field for the future. And I mean, I realized that the challenge to try and explain how out there in the world, development is a thriving, flourishing field, and developmental neuroscience is one of the sort of advanced frontiers of neuroscience. Well, so I realized then that PhD students come in fairly decided in what they want to do, because, hey, after all, an intelligent mind is not going to be a blank slate, right? It is going to decide what its interests are. And by the time somebody's finished their master's, the patterning is a little bit done. I came in with a Wellcome Trust Senior International Fellowship, and that fellowship allowed the flexibility of hiring BSc qualified JRFs or, sort of BSc qualified project assistants to work in the lab. Unlike the DBT, DST grants and so on, where all of these categories require MScs. So I discussed with Veronica, and she supported my starting a Masters by Research program at TIFR. Okay. And this really allowed my lab to take off. So in fact, I would like to now share screen and introduce my first two master's students, because of whom my lab started. So these are the first two women students in vertebrate developmental neuroscience in India, Sharada Bulchand and Aditi Vyas Naughate. Aditi now works with Nicholas Piramal, and Sharada is a faculty member in Singapore. Now, right BSc qualified students who are willing to try out something new joined our MSc program, and today our Masters by Research program is one of the most attractive, flourishing master's programs in the country, simply because we give them 6 months of coursework that equalize differences between people who come in with, we take in people with backgrounds from chemistry, even math, or any background who want to do a master's in biology. And we find that these students are willing to take risks and explore and invest in their own careers, because ours is a 3-year program and it's a solid research degree. So we have students who don't know what they want to do further in life, and that's fine. We welcome them because it's okay to be young, and it's okay to be undecided. At some point in life, one is undecided. We have students who know they want to do research, but they want to taste what research is like before they decide what they want to research in. And that's fine. Our master's program is for them. We have students who know they want to do a PhD, but perhaps not in India. They want to go abroad. And that's fine. Nobody should be restricted in where they should work. We don't want to trap people in, although there is an option for them to stay on to do PhDs. We say, fine, we'll train you. We'll show you what research is like. You will contribute 3 years of your bright young minds to Indian science, and then you go wherever you wish. So it's a win-win situation, and that's why this program is thriving. And what I'm trying to showcase is that the kind of support that people like Veronica gave to start this program, and the kind of sort of creative impetus that we got to shape it, is what allowed a thriving research program to succeed, not just for developmental neuroscience, but for other fields. So I'm just going to show you how for the first maybe 8 years of my lab, all the papers I published were first authored by a master's student. Okay, so this is Ryan Remedios. He had first author papers in Nature Neuroscience and Journal of Neuroscience, and he's now a faculty member in Munich at the Helmholtz Center. This is Vanisha Lakhina, a real bright spark. She's just finished up her postdoc in Princeton. This is Mugdha. Mugdha was a collaborative student with Veronica and me, and we actually looked at mouse genes in the fly, making a transgenic fly with some mouse construct. This is Aditi Falnikar. This is Lakshmi Subramanian, who started as a master's student and then continued as a PhD student, and is one of the most creative minds I've met. So I just wanted to say, that these are the sort of young developmental neuroscientists that I've had the privilege to start my career with. And so this was 99. Shortly after I returned to India, Shamala Mani returned too, and she set up her lab at the NBRC. So I'd like to now showcase Shamala Mani. Here she is, a lovely picture. Shamala started her career at the NBRC in 2000, so just a year after I did, and she and I were, long distance buddies for a long time. We exchanged reagents. She would, every time she'd fly through Bombay because their animal house wasn't set up, she would take mouse embryos on ice with her in a thermos. And we maintained a lab database of all the probes we had and all the constructs we had so that we could share them. By the way, this is an aside, but one thing about a small community is people help each other. So all of my mouse strains are cryopreserved, sperm cryopreserved at the facility at NCBS. And obviously you'll have to get permission and sign the appropriate MTAs. But if anybody would like to have the mice that I have taken the trouble to import and breed and so on, you're most welcome to have them. And you can write to me anything I've published on, and I will let you know whom to contact for the permission. I believe reagents should be shared because that's the only way we can all help each other get ahead. So routinely, the few mouse development labs share Cree drivers and so on with each other. And this is an open offer. I will help in any way I can. Okay, so Shamala moved from NBRC to IISC and is now in Paris. So she ran her lab she had a long run of 16 years in which she produced some very interesting work in the cerebellum. Okay. So, sorry, I'm trying to dance around because my windows are being occluded here. This particular photo was taken when Shamala won an award, and I'm just going to read the citation because it makes me so happy. Okay, Shamala Mani, Associate Professor at the Center for Neuroscience, Indian Institute of Science, at the investiture ceremony organized by the Consul General of France in the Alliance Française in Bangalore on June 14, 2010. Dr. Mani has been awarded the Chevalier de l'Ordre de Palme Académique, the Knight of the Order of the Academic Palms. The French government gives this prestigious award to individuals recognizing their distinguished contribution to academia and education. So hats off to Shamala. And here are a few studies that I'd like to showcase. This is Shamala's very early work. So when Shamala was a PhD student in the Mary lab, she was instrumental in actually making the GAP43 knockout. This was really her hard work. And then in her own lab, she found that this protein, GAP43, is key to mitotic spindle control and centrosome-based polarization in neurons. So this was a very cool early paper from her lab. And then she continued her interest focusing on the cerebellum. So here, this study shows how asymmetric cell division of granule neuron progenitors in the external granule shows asymmetric cell division in these progenitors in the EGL of the mouse cerebellum. Okay, Parthiv, so one thing about being a small community is we know each other's students. Parthiv has spent some time in my lab and he is now finishing up a postdoc with Kathy Millen in Seattle. So this is a nice integrated community. Subhashika finished her PhD in University of Geneva with Denis Chabourdon and published a very cool paper there and has now moved on. Okay, now I'd like to showcase a couple of papers that Shamala published when she was at the NBRC. Okay, this is about sonic hedgehog in the cerebellum.
Shubha?
Yes.
Hi, I'm so sorry to interrupt you, but we're running a little over time. So
I'm sorry. Yeah, I have just two more slides. Sorry.
Thank you. Yeah.
So this is about sonic hedgehog in the cerebellum, and this is the human cerebellum. And here I want to showcase that Shamala collaborated with another developmental neuroscientist, Sonia Iyengar, who is the next person I'm going to showcase. Sonia, who's at the NBRC, started with an interest in the axonal arbors of song learning in zebrafinch and has now moved to studying the human auditory cortex and sort of maintains a broad interest because when I looked at her recent papers, I found this one about the role of opioid receptors in song learning. So she's had diverse interests. And now I'm showcasing women neuroscientists, but it's not like there are a whole lot of men out there whom I'm ignoring. Okay, so I think there is one other developmental neuroscientist, Anindya Ghosh at NBRC, whom I'm not showcasing because I'm focusing on women. But it's not like these are, a few women amongst a whole category of men. These are just a few neuroscientists, and most of them happen to be women. Okay, and here's the last and sort of youngest neuroscientist in the history of neuroscience I'm going to showcase. And there's an interesting story about her that I would like to take a little time to tell, because this is inspiring. Okay, Jonaki Sen started her lab in 2007. So, and she had a background in retinoic acid signaling. And here is an early paper from her lab. And here she moves to mouse and looks at, so while she started with retinoic acid and has later on continued with Wnt signaling, here she's moved to mouse and has looked at BMP signaling in polarity and dendritrogenesis. So what I'm trying to say is that people have diverse interests, or successful people seem to have diverse interests, and that seems to be a very good thing, to not have just one focus, but to keep multiple projects going. And this study is a really cool study. I was privileged to be the external thesis examiner of Sovimal Sindhu. Okay, they found, sort of in parallel to my lab's early work, showing that the Wnt expressing cortical hem is like an organizer that induces the hippocampus next to it. They found a microRNA that normally restricts Wnt7b in the chick. Okay, and that if this microRNA is removed, Wnt7b shows up ectopically and literally induces ectopic hippocampal phage in the chick. So this was extremely cool, and much credit to Sovimal. But the story I want to end with, the sort of inspiring story I want to end with, is Ande Ka Fanda. Okay, so much of Jonaki's lab's work is on the developing chick. Now, when Jonaki and Amitabh Bandyopadhyay, who works on bone development in chick, moved to IIT Kanpur, they did not have a supplier for chick eggs, for fertilized eggs. Now, what to do? I mean, I spoke of having problems having an animal house. Well, they didn't have access to chick eggs. So in a remarkable, sort of inspiring feat of literally grassroots level effort, they trained somebody who did not know poultry farming. Okay, oh, I guess I closed the window. I'm just going to talk. They trained somebody who did not know poultry farming, who in fact was part of their department's cleaning crew. Okay, they invested 50,000 rupees in his sort of effort, sent him to train in how to do this to the Central Avian Research Institute, and basically helped him set up poultry farming so that he could start a business. Okay, and because of this business, he now sends his kids to school and, has become, uplifted from his previous situation. And his business provides the Bandyopadhyay and the Sen labs 120 eggs per day. They get a fresh supply of fertilized eggs every day at a rate that allows him to maintain a healthy income and so on. And this is fantastic. I mean, this is capacity building, par excellence. And I think this example really needs to be written up in a blog and publicized. So this is why I wanted to showcase this story. And I'd like to end with an encouragement to everybody, saying, we do research, and that's fantastic, but we shouldn't lose sight of the fact that our research is funded by the taxpayer and has to be shared. So research is not fun unless it is shared, and indeed the taxpayers who fund us sort of own the fun. So outreach is important, and to this end, I've had this other outreach dimension to my activities, which I'm happy to share with people who want to know. This is the ribbon game, in which I literally connect up an audience with ribbons along the lines of the visual pathway, the what and the where and so on. It's kind of fun, and it's online, so people can catch it at YouTube. And this was a TED talk I gave in Hindi about critical periods, hosted amazingly enough by Shah Rukh Khan. Any avenue to outreach is important. And even as we are all stressed with our research and so on, we shouldn't hold back on the outreach. So that's a message that I would like to end with. And thank you very much for hearing me out. And sorry about the extra time. I got a little carried away, but I hope it was interesting and useful.
No worries. And thanks a lot, Dr. Tole, for taking us on a journey of the field. Very nice insights. Thank you. Okay, so let's extend a warm welcome to our next speaker, Dr. Sonia Sen. So Dr. Sen is a senior scientist at the Tata Institute for Genetics and Society, Bangalore. And she runs the Neurodevelopment and Behavior of Drosophila and Anopheles program. where she and her team work on understanding the gene expression networks that control how neural stem cells diversify and lead to the development of a functional brain and behavior in Drosophila fly and Anopheles mosquito, respectively. And she focuses on applications such as reverse engineering, the development of specific neuronal cell types from neural stem cells. She's also a recipient of the 2019 Ramalingaswamy Fellowship, the 2015 Fulbright Nehru Postdoctoral Fellowship, and is a member of the Early Career Advisory Group for ELI. So today, Dr. Sen is representing the neuronal development field and will give a talk on understanding the molecular basis of insect brain development that diversifies stem cells to produce functional neural circuits and behavior. So Dr. Sen, thank you for accepting our invitation to talk on this conference. And the floor is all yours.
Okay, thank you, Anupama, and thank you, Shubha, for the really wonderful overview of neurodevelopment in India. I'm going to go ahead and share my screen. So can someone please confirm if you can see my screen and hear?
We can hear you. It's just a blank screen right now.
Yeah, that's perfect. That's how I wanted to start. Okay, so yeah, okay. So yes, so thank you, BiasWatch India, for doing the really important work that you do, and to your volunteers for helping you do that work. And thank you for organizing this wonderful conference that has allowed all of this showcasing of the wonderful science that women in India do, neuroscience that women in India do. And I hope that what it'll do is inspire more women to take on science and I mean, essentially, it would be a shame if we lose 50% of our intellectual capacity. And I hope that this effort will inspire more women to take on science. Okay, so I will jump straight in and talk about the work that we do in the lab. As Anupama said, I work at the Tata Institute for Genetics and Society, which is a center at INSTEM, the Institute for Stem Cell and Regenerative Medicine in Bangalore. And there I've set up the Neurodevelopment and Behavior Group, and we basically run two programs over there. The first is to understand how neural diversity is generated in the brain, and we use Drosophila for all of these studies. And the second is to understand the neural basis of blood feeding behavior of mosquitoes. And for this, of course, we use mosquitoes, but we also go back and forth between flies for this part of the program. Now, these two programs seem quite different, but they are stitched together by one central concept that bring them both together. And that is that for both of these programs, we don't worry about either diversity or behavior at the level of individual neurons, but we worry about it at the level of neuronal stem cells and their lineages. So let me explain. So I'm going to start by showing you a picture of the brain, of the fly brain, the seemingly simple fly brain. So this is an image of a 3D reconstruction of 50% of the neuron of the adult fly brain. I use this image to illustrate how even the fly brain is so complicated that it looks essentially like a mess. So, and if you're worried about understanding the brain or the behavior that it produces at the level of these individual neurons, it's quite a daunting task. But what we've learned in the flies is that actually all of this complexity is put together by very discrete units that make up all of this complexity. So this is one such unit or module that makes up that complexity. Here's another one, and here's a third one. So each one of these modules consists of a cluster of cells, say 150 or 200 of these cells that cluster together. They put their axonal projections together and innervate similar regions within the brain. And what we've learned is that each one of these modules, this one or this one or this one, is constructed through a single neural stem cell. So here's an image showing you in the developing larval brain. Here's, I hope you can see my arrow, but here's a neural stem cell, which is in the larval brain, which has generated a bunch of neurons. Now, if you wait for this to develop through pupil life, it will elaborate into a module that looks like this. So this is the first important thing that we've learned from the flies, that it's constructed through developmental modules. The second important thing that we've learned from flies is that and you'll see it in this image, each one of these developmental modules, so for example, this one over here seems to be innervating a similar structure. This one is innervating the antennal lobe that Shubha spoke about. Now, because it's innervating the antennal lobe, we know that it's processing olfactory information. So this is the second important thing that we've learned, that these developmental modules that put together the fly brain are actually also functional units in the brain. Now, if you think about the brain, and not worry about it from the perspective of individual neurons, but from the perspective of these developmental modules that make up the functional modules of the brain, then you've scaled down the order of magnitude of your problem. You're not thinking about 150,000 neurons, because that's how many make up the fly brain. You're thinking about 150 such modules. Much more tractable. And so this is the central theme that ties both of our programs together. We worry about development of diversity and function at the level of neuronal stem cells and their lineages. Okay, so since we're talking about stem cells and their lineages, let me tell you a little bit about how they form. So in the fly, all neuronal stem cells are set aside extremely early during development, even as the embryo has just finished gastrulation. And on the ventral surface of the embryo is a sheet of cells that gets specified as the neurectoderm. And the neurectoderm now on it can form the neural stem cells. Now, because this event is happening so early, it is coincident with the embryo actually being patterned in its anterior, posterior, and dorsoventral axes. So you can imagine the embryo with its ventral surface as the neurectoderm experiencing all of these APDV patterning genes, such that if I were to turn this embryo around and show you what the neurectoderm looks like, here are neural stem cells forming on it. But remember, the neurectoderm is experiencing all of your APDV patterning genes. This is engrailed, an AP gene over here. If you superimpose these two images, what you'll immediately see is that a stem cell that forms within this engrailed domain is molecularly distinct from its neighbor, which is probably coming out of a different AP gene. So I'm showing you one example over here, but of course there are very many APDV patterning genes. And so this is schematized over here. Each circle represents a neural stem cell that's forming on the neurectoderm, and the neurectoderm is represented by these APDV patterning genes, engrailed over here, gooseberry over here, MSH, the DB patterning genes, and other DB patterning genes. And so what you can immediately see is that as the stem cells are forming, they're experiencing unique molecular environments. So 7-4, the neuroblast 7-4 over here, is experiencing engrailed and MSH, whereas 5-6 is experiencing gooseberry and MSH. And so this is essentially where spatial patterning of the neurectoderm takes what would have been a homogeneous pool of stem cells and imparts unique molecular identities onto them. And now what this in turn means that unique molecular identities of stem cells can translate into unique lineages and circuitry within the brain. And so this stem cell lineage over here, all of these neurons made from a single neural stem cell has this innervation pattern, whereas this stem cell lineage over here has a totally different lineal output. So what I'm telling you is that spatial patterning seems to lie at the heart of how neural diversity is generated within the fly brain. Other brains as well, but we're talking about the fly brain. That's not to say that spatial patterning is all there is. Spatial patterning brings about interlineage differences, makes each stem cell different from one another. But there's something else that stem cells use to generate further diversity, and that's temporal patterning. Now, temporal patterning I will not talk about it too much, but a lot of work has been done on temporal patterning. It's essentially a set of genes that are expressed in series within the neural stem cell as it's dividing and generating neurons through development. Now, the thing about temporal patterning is it's the same across, if not all, majority. There are a few temporal series that are expressed in many, many neural stem cells. And because they're the same in spatially distinct stem cells, we believe that spatial patterning is what provides temporal cues, the context to make lineage-specific neurons. And so we focus on spatial patterning of neuronal stem cells. Okay, so I'm going to step back a little bit and say that everything that I've told you about how stem cells are made different from one another is a tiny, tiny fraction of what we know actually happens during neurogenesis. All of that happens in the neurectoderm, right? And that's like 20 minutes of neurogenesis. So as soon as they form on the neurectoderm, the neural stem cells actually leave the neurectoderm. They pop out of it and into the embryo. So they've essentially lost that entire carefully constructed Cartesian coordinate grid system that they were experiencing. So they don't have that spatial information anymore. And yet it's not as if a stem cell, once it leaves the neurectoderm, begins to generate neurons that are not lineage specific. An olfactory lineage will still make olfactory neurons, and a central complex lineage will still make a central complex neuron even after it's left the neurectoderm. So how is it that that happens? How is it that after leaving the neurectoderm, these stem cells still retain their spatial identities? So there are two ways in which you could think about this. One is that maybe whatever it's experienced in the neurectoderm gets somehow imprinted as memory within the stem cell, that it remembers what its spatial identity was. And as soon as you're talking about memory in a developmental context, you have to invoke the idea of a chromatin landscape. And so I actually tested this idea during my postdoc, where I focused on two neural stem cells that sit right next to each other. The 7-4 over here, which arises out of the engrailed domain, and 5-6 over here right next to it, which arises out of the gooseberry domain. And what I decided to do was look to see what their chromatin was like after they left the neurectoderm. I was expecting to see differences in chromatin. I'm sorry, just to step back, both of these lineages, though they sit right next to each other, they make very different lineages. So 5-6 from the gooseberry domain makes a fairly small lineage with some glial cells and some interneurons. 7-4 actually makes a much, much larger lineage with these channel glia and actually interneurons, which this image is not doing justice to, which span the entire length of the neurectoderm. And when I looked at their chromatin landscapes, what I found was that once they've left the neurectoderm, these stem cells in vivo, remember, we're not taking these out of context, they're not ex vivo, we're assaying chromatin from these individual neural stem cells in vivo, and we find them to be dramatically different from one another. So yes, stem cells do have different open chromatin landscapes once they leave the neurectoderm, but does that correspond to what the temporal cues read in that chromatin landscape? You'd expect, if this were true, that the chromatin, that where the temporal factors bind should be different. And so the primary temporal series that acts in the embryonic, developing embryonic brain is actually this one. And of course, I didn't look at all of these. I picked the first of the series, which is hunchback. And so I looked again in vivo of hunchback binding within these two identified spatially distinct stem cells. And what we found was that hunchback bound very, very different loci within these two stem cells. So just to show you the top few of those as an example of how differently they bound, these guys are loci that are bound by hunchback in 5-6 alone and not in 7-4, and vice versa. These are bound by hunchback in 7-4, but not in 5-6. So, okay, chromatin landscapes are different. What the temporal factor can bind to and therefore regulate is different. But that's not all what this idea would call for. This idea would call for the chromatin landscape actually guiding where hunchback binds. So you would expect that that in 5-6, where the chromatin is open, is where the temporal factors will bind, and likewise for 7-4. And so this is what we did. We picked all of these sites, and not just these 5, but all of the ones that were enriched for hunchback binding in 5-6, and we compared the chromatin at those loci in 5-6 and 7-4. And that's what I'm showing you over here. So stacked up over here are all of those loci. And in the signal, we're showing you chromatin binding. The blue represents the chromatin of 5-6. The green represents the chromatin of 7-4. And you can see at the loci where hunchback binds in 5-6, the 7-4 chromatin is basically closed, and the 5-6 chromatin is nice and open. You can do the converse and look for the loci that are enriched for hunchback binding in 7-4, and look at what the chromatin are like in these two stem cells. You notice over here that 5-6 chromatin is closed at those loci, and in 7-4 it's nice and unfurled. And this is just a sanity check that if you look at similarly bound loci, the chromatin is similarly open in those two neural stem cells. So what do we think is happening? Clearly, the chromatin is different, and that's guiding what the temporal factors can read. But what we think is setting up those chromatin differences are these spatial patterning genes. So because the spatial patterning gene in 5-6 is different from the spatial patterning gene in 7-4, we think that they're perfectly positioned to set up these chromatin landscapes and drive the future identity of the neural stem cell. Okay, so that's one way in which we think the spatial identity of a stem cell can be maintained even after it's left that Cartesian coordinate system. But there is another possibility, which is that actually it need not be via the chromatin, but instead that molecular identity needs to be maintained by factors that continue to be expressed within those neural stem cells. And actually, people have tried to get at this idea. Does this actually happen? So this is work from Gerhard Teschnau's group in Mainz, and they did this painstaking effort of finding antibodies against those early patterning genes and mapping each neural stem cell with respect to each one of these genes. And then they released these beautiful maps, which people across the world, including myself, picked up and said, okay, do any of these actually correspond to things that maintain identity of a stem cell? And unfortunately, if it did, what you'd expect is that you would be able to fiddle around with identity, right? Unfortunately, when people did these type of analyses, and there have been many such studies, none of them actually spoke to identity. We uncovered a lot of very important processes, genes you heard from Sonal yesterday, genes that are involved in proliferation control, very important for brain development. The other type of phenotypes that were uncovered were targeting defects, so neurites that would spill out of their domain of innovation, or didn't innovate it sufficiently, or overshot a target that they needed to, or didn't reach the target that they needed to. So targeting defects. And the third category of phenotype that people usually uncovered when they did this sort of analysis was finding non-neural cells. So they would, these neural stem cells would generate cells, progeny, which wouldn't be neurons or glia. It was quite by chance that I uncovered during my PhD a factor that actually spoke to the concept of identity. So I was studying the antennal lobe and the neural stem cells that constructed the circuitry within the antennal lobe. And I was studying the genes that were involved in making sure that those antennal lobe circuitry were constructed properly during development. So I guess this was the next phase of Veronica's neurodevelopment. She did a lot of work initially on the peripheral circuits, and then later on, she began to look at the central circuits, and this was part of that effort. And while I was doing that, I noticed that one of the factors that I was studying was also expressed, it was called OTD, it was also expressed in a lineage which was not contributing to the antennal lobe, but it was contributing to the central complex. In fact, it avoided the antennal lobe almost completely. No, absolutely, completely. It didn't actually innervate the antennal lobe. And because I was so familiar with the antennal lobe, I realized, and I was using tools that were random and stochastic, in some of my experiments, I would find this lineage, which I wasn't interested in, would lose OTD. And when it did, I found something wonderful, that it converted to an antennal lobe lineage, one of the ones that I was studying. And you can see the similarity in innervation pattern between these two. Now, we went through great efforts to show that this was actually a transformation and not just this lineage going wonky. And I won't tell you about that, but believe me when I say that this was a true transformation of this lineage into this lineage upon the absence of one gene from the neural stem cell. Not only did it look like an antennal lobe lineage when it transformed, it changed its molecular identity, it changed its neurotransmitter identity, and remarkably, when I puffed odors at the antennae of these brains and I monitored calcium activity in the antennal lobes, this lineage, which normally would not have seen olfactory information at all, began to respond in the glomeruli exactly like this lineage would have. So all of this to say that quite by chance, in an embarrassing effort of looking under the lamppost, I uncovered that yes, neuroblast identities do indeed need to be maintained by factors that are expressed in them throughout development. And what this in turn suggested was that we are yet to uncover what the transcription factor codes are that maintain these identities. We don't know what they are. And so this is the task that we've set for ourselves in my lab. We want to discover what the spatial factor codes are that diversify and maintain the stem cell identities and therefore the circuitry within the brain. And the reason we think that now is the right time to do this is because of the advent of single cell transcriptomics. Because now you are not restricted by the handful of antibodies, good, bad antibodies against a small number of transcription factors, but you can actually assay the entire transcriptomes of individual neural stem cells. And so this is what we're doing. And of course, this is work in progress. So I hope that the next time NeuroFem happens, I'll be able to update you on our progress. Okay, so I'm going to switch gears here now and tell you a little bit about our mosquito neurodevelopment and behavior program. You may or may not know that mosquitoes, both male and females, actually usually drink nectar or sap. And it's only the female, and even that only sometimes, that she switches to taking a blood meal. And so this simple but dramatic change in dietary preference lies at the heart of an enormous number of diseases that mosquitoes are able to carry. Closer to home, we have dengue outbreaks and chikungunya outbreaks regularly in Bangalore. Bangalore doesn't have malaria, but India certainly does bear the burden of malaria. So mosquitoes really do transmit a whole lot of diseases through this simple behavioral choice of dietary preference. And so we're interested in the neural circuits that drive this change in dietary preference. So just like neural circuits have the ability to sense your external environment and mount a behavioral response that is appropriate for that external stimulus, just like that, circuitry have the ability to sense internal physiological states, and then mount a behavioral response that seeks to satisfy that internal state. And so the things that we think could be signaling that internal state are the ovary or oogenesis. And this is because the one thing that the female mosquito definitely needs blood for is oogenesis. Without blood, a female mosquito cannot make eggs. So we think that the ovary might be well placed to signal to circuitry to drive this change in behavior. And another possibility is mating. You heard yesterday from Pinky how mated females have an altered dietary preference for salt. So she was talking about mating with respect to salt intake. We're interested in mating with respect to blood feeding. So here's the task that we've set for ourselves. We want to map the mosquito brain. Once we do that, we want to map the blood feeding behavior onto the mosquito brain. And once we've done that, we want to be able to ask how internal states, as signaled by whatever we find, oogenesis or mating, or both, intersect with the circuitry that we've identified to drive this behavior. Okay, I will acknowledge over here that the scale of the program that we're proposing is massive, but I will say two things in response to that. The first is I'll flash back at you this title slide of ours. Remember, we're not interested in any of these things, in mapping the blood feeding behavior, mapping the brain. We're not interested in doing this at the level of individual neurons, at least not yet. We want to do it at the level of neuronal stem cells and their lineages, because we think that that's a good place to start so that you can get a handle on the problem and you can always look at individual neurons later. Okay, the second thing that I want to say is that I don't actually envision this as a program that me sitting in my lab with my colleagues of 10 people are setting out to do. We actually envision this as a consortium level effort. We will put in our efforts to show proof of concepts, and then we want to open this up as a consortium that brings people together with diverse expertise so that we can really tackle this problem at scale. Okay, so just to give you a feeling for the kind of approach that we're taking, remember we want to map the brain and map blood feeding behavior onto the brain. So we want to do it in two ways. We want to do it molecularly as well as neuroanatomically. Our molecular approaches are essentially to take the brain, the mosquito brain, to dissociate it into single cells like this, and then take it into single cell sequencing platforms. And the reason we're doing this is because in the fly, when you do this with the adult brain, you get these clusters, these molecular clusters that correspond to lineages within the fly brain. And in the fly brain, because you know so much about these clusters, you can actually go back and pinpoint a particular molecular cluster to a lineage. So we want to do this for the brain in general, but also to do it under conditions that will allow us to pinpoint the molecular clusters corresponding to lineages that are responding to blood feeding behavior. Okay, but molecular clusters don't tell you anything about circuitry. For that, you really need neuroanatomy. And so this is the other thing that we're going to do. We want to be able to I showed you that slide from Shumin Lee's lab with all of those beautiful brain lineages labeled, individual neural stem cells and their lineages labeled. That's what we want to do for the mosquito brain and map each one of those lineages in this manner. So to do this, you need a way of sparsely labeling individual neural stem cells. If you have too many labeled, it's a mess, like I showed you earlier, and you don't know you can't decipher a module there. And you want to do it stochastically such that if you were to survey enough brains, you would have surveyed all of the lineages within the brain. And because we're going to be working with a non-model organism, we want very clean, crisp genetics, not the messy genetics of flies where you sometimes have 14 transgenes to be able to do clonal labeling like this, but we want to do it with a single transgene. And this hasn't even been done in flies. So what we're doing right now is standardizing the logic of how we're doing this in flies. And as soon as we've got the logic back, we have the right frequency of stochasticity and sparseness, we will take it into the mosquitoes, and hopefully when we open up those brains, we'll have these brains with lineages labeled, few lineages labeled such that we can map them. And now you can imagine, if you have the ability to do this, you can combine this method with reporters of neuronal activity, with factors that can allow you to manipulate neuronal activity through light, temperature, chemicals. And so through those approaches, you'll be able to map lineages that are involved in the blood feeding behavior. Okay, so again, all of this is work under progress. We have the ideas down pat. We have our approaches. We're building our tools, testing which parts of all of these work in the mosquito, what needs to be tweaked to be to get it to work in the mosquito, and we're trying out our logic in the flies. So again, hopefully in the next NeuroFem talk, I'll be able to give you some updates on how this is going. Okay, so I'm going to stop because I think my time is up, and say thank you for taking time on a Sunday to come and listen to our work. A big thank you to the people who fund us, Tata Trust and Ramalinga Swami Fellowship. This is the gang that does all of the mosquito work. So Roshni and Mandar there drive all of the neurodevelopment part of it. Pooja and Prashali are driving the behavior part of it. Varun was an intern who's now in Scandinavia doing his master's, but he was involved in a lot of the design of the Drosophila constructs. Lakshmi has been moonlighting during her BSMS at IISER Kolkata, doing some computational work at identifying enhancers in mosquitoes, but she's going to be doing her master's thesis with me starting later this year, where she's going to jump ship and join the Drosophila neurodevelopment gang, which is led by Ayanti, Sonali, Hema, and most recently Bhagyashri. And finally, something that I didn't even mention is a program that we have on evolution of the brain, where we're looking at a new phylum, a recently described phylum of Xenocoelomorpha, and that is being led by Varnika. And finally, a big thank you to the Bangalore Life Sciences Cluster, of which we're a part. To be able to take on 2 programs of this scale is only possible because of a place like the Blisk campus. And it's wonderful to be a part of it. With that, I will stop and say thank you, and I would be very happy to take any questions if there are any.
Thank you so much, Sonia. There are quite a lot of questions. That was a fascinating.
Oops, I'm sorry, I can't hear you. Is there? Sorry, can you? Sorry, Anupam, I couldn't hear you for a second. Could you say what you were saying?
No, I said fascinating talk, and thank you so much. And there are quite a few questions, so I'll try to field some of them. Yeah. So one from Poonam Thakur: is it possible to do awake behaving recordings of mosquito neurons while they're feeding on blood?
So, if it's possible, we're not the ones doing it, but what we've planned is things that report neuronal activity, which you can look at post hoc. So something like Campari, where you can expose mosquitoes while they're behaving. We'll have to standardize a lot of that, but yes, you can expose mosquitoes to a certain wavelength of light while they're behaving, and then open up their brains afterwards, where you can see a photo conversion from green to red and identify which lineages are active.
Okay.
Yeah, thank you for the question.
Shreyas Gadge wants to know if the spatial differentiation of stem cells translates to topological differences at a network level. Is there a way for topographical manipulation to find out effects of patterns, etc., on neural diversity?
So topological differences of, can you read that phrase again? Topological differences of?
At
circuit level.
At a network level.
I'm not quite sure what you mean by that, Shreyas, is it?
Yes.
So the spatial differences that I'm talking about, Shreyas, is a developmental coordinate system of where that stem cell is being formed. So maybe what you're asking, I'm not sure if this is true, if I now were to move a stem cell from one spatial location to a different one, would that create a different stem cell identity and therefore a different circuitry? If that is what you're asking, then yes, those are the kinds of things that we will test. The advantage of doing this in flies is you don't have to, although you can, and people actually used to do things like this, you don't have to physically take out a stem cell and put it somewhere else. You can do genetic manipulations which effectively simulate that same experiment. But I'm not sure if that was your question.
Yeah, I'm sure. So you should also be able to, in case, they may be able to get in touch with you in the chat box.
Yeah, sure, definitely. I will look up your chat.
So we have time for one more question. Abhilasha wants to know if the timescales of behavioral changes observed in mosquito feeding preference aligned with changes in stem cells and the lineages. Should the approach depend on this timescale?
Should we what? Can you read that again, Anupama?
Should the approach depend on this time scale?
So, Abhilasha, we're going to be assaying all of this after we're going to be assaying behavior after all of the development is done. So the circuitry will all be in place, and then we will assay the behavior. So we don't really need to worry about the time scales of the development of the circuitry.
Okay, there are a few more questions in the question and answer box, so it would be great if you could answer them.
I'll do that. Yeah.
Yeah. Thank you so much.
Thank you
so much for the wonderful talk. Yeah. Okay, so it's now time for our third invited speaker of the day. So please extend a warm welcome to Dr. Nivethida Thirugnanasambandam. She's a Wellcome Trust DBT India Alliance Intermediate Clinical Research Fellow at NBRC, Manesar, where she runs the Human Motor Neurophysiology and Neuromodulation Lab. She and her team work on understanding the physiology of surround inhibition in human motor cortex and the pathophysiology of movement disorders such as Parkinson's disease, levodopa-induced dyskinesias, and also how physiological pathological alterations in brain states can impact the outcomes of neuromodulation. She's won the 2020 Har Gobind Khurana Innovative Young Biotechnologist Award, is a member of the Indian National Young Academy of Sciences, and was also awarded the DBT Ramalingaswamy Re-Entry Fellowship in 2017. So today she's representing the systems and computational neuroscience field, and will give a talk on understanding how non-invasive brain stimulation has enriched our knowledge in the pathophysiology of movement disorders and its potential diagnostic and therapeutic implications. So Dr. Nivethida, thank you so much for accepting our invitation, the floor is all yours.
Yeah. Thank you, Anupama. Can you hear me? And can you see me?
We can hear you and we can see your slide. Yeah.
My slide. And do you see the cursor moving?
Yes.
Yeah. Okay, great. So thank you, BioSwatch India, for the opportunity. And yeah, as Anupama said, I'm an India Alliance faculty fellow at the National Brain Research Center, Manesar. And today my talk will be on probing the pathophysiology of movement disorders using non-invasive brain stimulation. So we are basically switching gears from flies to humans and from development to patient research. So I don't have these really colorful slides because our access to the human brain is very much limited even today. And but I will tell you what is possible at with the technological and scientific advancements of today. So by pathophysiology, I mean abnormal functioning, which means here specifically talking about abnormal functioning of the brain in patients with movement disorders. And I will, through my talk, I will take you through what movement disorders are and what non-invasive brain stimulation actually is. And I will also tell you about how we can use non-invasive brain stimulation to study an alert active brain in humans as the disease progresses, and what information we can get by using these techniques. Finally, I will close by saying what kind of work I am currently doing in my lab, which is still pretty young. Okay, to start off with, just a minute. So motor control is something that is indispensable for our daily lives, and we hardly recognize it, but it comes very naturally to us when we are just walking or writing or say eating and say pouring water or something. But we fail to realize the complexity of the system that makes all this possible. So a large portion of the brain is dedicated towards performing movement. And we probably don't even realize how complex the system is until we lose motor control, or we see somebody else who's lost motor control. So there's Professor Daniel Wolpert, who is a movement neuroscientist, clearly says that we have a brain for one reason and one reason only. And that's to produce adaptable and complex movement. There is no other reason to have a brain things like sensory, memory, and cognitive processes are all important, but they are only important to drive movement. And I pretty much agree with him. It's so as to, even evolution-wise, there is evidence saying that the earliest and the most primitive form of brain is seen in those organisms that have just started to move. So it could be for various reasons, for to escape predators, to search for food, or for mating, and for many other reasons. But the whole brain works in synchrony to produce adaptable and complex movement. And what happens when we lose motor control? So that is when we have movement disorders. There are different parts of the brain that control movement may get affected, and these are some examples of certain movement disorders that are pretty much common. So the first one is tremor, which we have probably seen more commonly. It's a rhythmic shaking of a body part, usually the hand. The second one is Parkinsonism. We've probably heard of Parkinson's disease, say in our family, or maybe in a family of our friends or relatives. And third one is called dystonia. It's abnormal posturing of a part of the body, in this case, the neck. And the last one is chorea, or dance-like writhing movements that occur in certain patients. This is a patient with Huntington's disease. So there is some kind of abnormality that is happening in all these disorders. And movement disorders are a class of neurological syndromes which are characterized by abnormal, involuntary movements or postures. So when I say abnormal, it could be either excessive movement or it could be a decrease in the normal movement. And and movement disorders basically constitute about 10 to 15% of the patients you would see in a neurology clinic. And the most common movement disorders are essential tremor, Parkinson's disease, and dystonia. One common feature of all these movement disorders is that the pathophysiological mechanisms that underlie these phenomenology in the disorders are still largely unknown. So we know that the pathology lies in, say, the basal ganglia or in the cerebellum, but then we do not know why there is such varied phenomenology that is seen, even say in a subset of Parkinson's disease. So some patients present with tremor, whereas some patient presents with akinesia or slowness of movement. We are still not able to understand the exact pathophysiological mechanisms that underlie these disorders. And therefore, we are not able to come up with treatment strategies that would be effective in these patients. So recently, or in the last few decades, neuroimaging studies, could be EEG or MRI, have given us some information about the altered functional connectivity in the cortical and subcortical brain networks in these patients. So we think movement disorders could be associated with abnormal connectivity between brain regions in these patients, and not just, it's a single neuronal level, or it could be a system problem. As of now, neurologists and movement disorder scientists believe that there is immense potential for non-invasive brain stimulation and non-invasive brain recording methods in understanding these pathophysiological mechanisms that cause movement disorders. So that is exactly what I will be focusing on. So there are two types of non-invasive brain stimulation methods that are currently in use. One is the transcranial magnetic stimulation, or the TMS, as I would call it. And the second is the low intensity transcranial electrical stimulation, which could be in the form of direct current or alternating current. So accordingly, we call it TDCS or TACS. So with TMS, what we do, so this is a picture where you can see the stimulator, here, the blue one on the left side, connected to a coil, which is the shape of the number 8. So these are basically two circular coils that are tied up together. And what this machine does is it's basically a capacitor that discharges current, very high intensity current to the coil here. And this rapidly changing electric field generates or induces a magnetic field that is perpendicular to the direction of the current. So what happens is this perpendicularly directed magnetic field passes through the skull and reaches the intracranial cavity. painlessly, and the brain acts as the secondary conductor. So wherein, and this rapidly changing magnetic field induces a secondary current in the brain tissue. So and this electric current, depending on the stimulation intensity, if it is high enough, will be able to activate the underlying neurons. So basically, if you put this coil over the motor cortex, or the region of the brain that controls your movement, say your hand muscles, and you trigger the pulse, then your hand muscles will twitch. So it's basically activating your motor cortex to make, activate the peripheral muscles. So you could do this to any region of the brain, activate any kind of neurons. But because the depth of the strength of the magnetic field dies down rapidly with depth, at the moment, we are not able to reach deeper parts of the brain. So it's only the cortical region which is accessible by transcranial magnetic stimulation. So that is the drawback. But still, we have a lot that can be inferred from this method. Secondly, we're coming to transcranial DC or AC stimulation, wherein we're putting two anode and cathode, like two electrodes on the skin of the scalp, and delivering a current, a really low intensity current of, say, in the range of 1 or 2 milliamperes. This could sound quite crazy, because how can you expect like 1 or 2 milliampere current to even reach the brain, cross the skull and reach the brain? But there is evidence that it does reach the brain, but on the other hand, it does not work like TMS, wherein it activates the underlying neurons. What it does is basically it changes the membrane potential of the underlying neurons and increases or decreases their sensitivity to activation. So it basically makes them more or less excitable. It's like putting the brain in an electric field, and therefore changing the membrane potential of the neurons. So we have this also. So I will talk about how these can be implicated in understanding the physiological mechanisms in an alert individual, healthy individual, and how we can understand the say abnormal functioning of the system in patients. So a little more deeper into the mechanism of TMS, on how we can derive information about the brain or the functioning of the brain using TMS. So when we give a TMS pulse to the scalp, what happens is, say you're activating certain set of neurons in the brain, and this they generate action potentials, which is, say, this is the motor cortex. And specifically of the hand motor area. Most of the studies use the hand motor area because this region is most superficial on this to access by the TMS from the scalp, and also you have a very good readout of the output, which is the EMG, the muscle activity from the hand muscles here. So you can just stick in surface electrodes on the skin of overlying one of the hand muscles, and then you can record what is called motor evoked potential. So you give a pulse, and then you see on your EMG something like this, a recording like this, which is called the motor evoked potential. And the amplitude, the peak-to-peak amplitude of this motor evoked potential seems to be a reliable indicator of the excitability of the cortical neurons. Of course, this is not a direct measure, this is an indirect measure, but still this is as good as we can get. But now I think we have some more, I will talk about more cortical correlates, parameters that can be recorded by EEG directly from the brain and not from the periphery. Yeah, so that is about a single pulse. What you can do with TMS is also elicit double pulses or paired pulses, like what is called the SICI or ICF, so which means short latency intracortical inhibition and intracortical facilitation. So you can activate selectively inhibitory or facilitatory interneurons in the motor cortex by using paired pulses. So here, when you talk about paired pulse TMS, you're giving a conditioning pulse of a certain intensity, and then a test pulse of a different intensity at different intervals, time intervals. So we see that at shorter time intervals, you will be able to activate the inhibitory interneurons, thereby resulting in smaller MEPs. And at larger time intervals, you would be able to activate the facilitatory or the excitatory interneurons, which results in larger MEPs. So how does this even, be useful in understanding disease conditions? That will be my next slide. But before that, let me also tell you, remind you about what happens. This is an illustration of what happens with transcranial electrical stimulation. So you put an anode and a cathode, as I said, and what happens is either depolarized under the anode, the neurons under the anode get depolarized, and those under the cathode get hyperpolarized. So the focality is something that we have not been, is really bad when compared to TMS, when it comes to TES. But this is very much a recently studied technique, but it seems to have some behavioral effects, definitely behavioral effects, which need to be optimized by further research. So how would you even know what changes are happening in the brain by using non-invasive brain stimulation? So in alert humans, we use non-invasive brain recording methods such as electroencephalography or EEG, MRI, which you're probably more familiar with, the magnetic resonance imaging, and something called magnetoencephalography, a cousin of EEG. So it's called MEG. So using all these techniques in parallel with non-invasive brain stimulation, what you can do is identify the sources or say the brain regions that have changed their activation patterns or their activity because of brain stimulation. And using these sources, you can also, and advanced analytical methods, you can also map the connectivity between these brain regions. And this can be done at different behavioral states. So say at resting state, or when the subject is performing a certain cognitive task, or say a motor task. So then you can understand which brain regions are involved in this task, or which brain activity or the connectivity of which brain regions are altered during a different brain state in an individual. Or you can also see how this connectivity pattern is different in patients with a certain disease like movement disorders or any neuropsychiatric disease for that matter. More recent approach is a multimodal approach. Rather than looking at these changes offline, say before and after stimulation, it would be really good to look at these changes that are happening at the time of stimulation. But then technological advancements, we had to wait for technological advancements that would enable us to do so, because EEG, as you may know, is recording activity in the order of microvolts, and the TMS that you're giving, the stimulation that you're giving, is in the matter of say 3 or 4 orders of magnitude higher. So how would you even look at the changes such as say at difference in this resolution of amplitude? So what we do is we can now combine TMS with EEG in the newer systems, where you can record the EEG activity while you're giving TMS pulses over a certain brain region. And then after a few, say 100 pulses, you can average the time locked activity in different brain, in all over the brain. And then you can actually, what you would see is these kinds of potentials, which the amplitude of which basically signify different physiological phenomena. So the advantage of using this is that you stimulate one part of the brain, but then you will be able to record changes that are happening in remote parts of the brain, which may be connected by long distance connections, and also look into the global impact on the brain, rather than a very focal change that is happening at the site of stimulation. So, and in addition to this, you would also be able to look at the changes in the oscillatory patterns. By oscillatory patterns, I mean the different firing patterns of the different brain regions. I will not go into details of oscillations, but that is another thing that you can look at using TMS EEG. Also, we've been able to do TES MEG, so with a combination of electrical stimulation with TMS or EEG as well as with MEG. But technical challenges still remain, because signal processing is not that easy when you have an active electrical current that is being delivered to the brain, and you're also recording electrical signals from the brain. So yeah. So now I will just shift to showing you how different paradigms, that is the paired pulse or the repetitive TMS paradigms, can be useful in understanding the physiological mechanisms that underlie, say, a motor control. So the first one is paired pulse TMS. As I already told you, giving a conditioning pulse and a test pulse to the motor cortex, in this case, at different interstimulus intervals. At smaller interstimulus intervals, you get smaller MEPs, and you can measure how much the change in the MEP is with this paired pulse paradigm. And at longer interstimulus intervals, what you get is a facilitated or an increased MEP amplitude. So this is a graph that shows you how the MEP amplitude changes as you change the interstimulus intervals. And what happens in patients with dystonia or in Parkinson's disease. So this graph shows that the empty circles are the recordings from healthy individuals, and the filled circles are the ones that are seen in patients with movement disorders. You can see that, say, there's no inhibition. So 100% is the control MEP amplitude. So that you see here. And what is plotted is the normalized MEP amplitude. So which means anything below 100 means inhibition, and anything above 100 means facilitation. So as you can see, healthy controls clearly show inhibition of about even 50%, whereas in patients, you see a clear loss of inhibition at specific interstimulus intervals like 2 milliseconds, 4, 5 milliseconds, and so on. So this means that the Parkinson's disease patients or dystonia patients, they have less intracortical inhibition as compared to their healthy counterparts. And some other studies which have looked at what medication actually does to this abnormal loss of inhibition is that levodopa, which is the standard drug therapy for Parkinson's disease, is able to restore this lost inhibition in these patients. So it is also, I think, important to know that this inhibition, which is a parameter, a neurophysiological parameter, also correlates very well with a behavioral measure, which is response inhibition, that is the action stopping. So those with less inhibition, they seem to be more impulsive. So behaviorally also, it seems to correlate very well in these patients. So another variant of a paired pulse TMS is a dual site TMS, where you're not stimulating the same brain regions, which is just the motor cortex, but you're giving the conditioning pulse to a different region of the brain to test how this particular region 1 interacts with region 2, which is usually the motor cortex. So here, I've taken the example of the two motor cortices, the left and right motor cortex, and then where you're measuring the output in the left hand. Okay, maybe it's the right hand. So it's the right hand you're measuring, you're giving the conditioning pulse to the right motor cortex, and then you're looking at the impact of the right motor cortex on the left motor cortex. And what you see is at specific interstimulus intervals from 5 to 10 milliseconds, you see a clear inhibition, that is decrease in the MEP amplitude, which signifies that around 5 to 10 milliseconds, there is a clear inhibition that is exerted by the neurons, neurons that are connecting, that is connecting the two motor cortices. So not just bilateral motor cortices, this same connectivity, cortico-cortical connectivity, can also be studied in different brain regions, like the premotor, motor, in the ipsilateral or the contralateral or ipsilateral regions, and also say the supplementary motor area, and also the parietal cortices. So this is a method to evaluate reliably the cortical connectivity in healthy subjects as well as in patients with movement disorders. This is one example of a study that has evaluated the cortico-cortical connectivity in patients with focal hand dystonia, which means writer's cramp, where this is in healthy subjects, where they've seen that there is an inhibitory interaction between ventral premotor and motor region, and another inhibitory interaction between the anterior inferior parietal lobule, AIPL, and the motor cortex. So the dorsal part of the IPL seems to have a facilitatory interaction with or facilitatory effect on the M1 output. So this is in healthy controls. And when they studied patients with focal hand dystonia, they found that there is abnormal, or maybe there is deficient PMV M1 inhibition and deficient DIPL M1 facilitation, although the AIPL M1 inhibition was preserved. So further, the authors went on to do something called the repetitive TMS, where you give repeated pulses of TMS at a certain frequency. It could be either low or high frequency, or in a certain pattern, with the aim of inducing neuroplasticity in that particular region of the brain. So they gave, they gave inhibitory TMS, RTMS to the DIPL. And what they found was this interaction between PMV M1 was restored in patients with focal hand dystonia. So this implied that by interfering with DIPL, you would be able to say normalize or restore the interaction, the connectivity between PMV M1 that is abnormal in patients with focal hand dystonia. Whether this is associated with symptomatic relief, that needs to be studied. So another good example of using repetitive TMS to modulate functional connectivity is this study by Papa and colleagues in 2013, where they gave repetitive TMS to the cerebellum on both sides of patients with essential tremor. And we know that essential tremor involves the cerebello-thalamocortical network. And they studied the functional connectivity in this particular network and another control network, which we call the default brain network. And after 5 days of stimulation, of a certain stimulation paradigm with RTMS, they found that the connectivity of the cerebello-thalamocortical network alone increased, whereas that of the default brain network did not. And this was clearly associated with improvement of symptoms in patients with essential tremor. And this improvement was not just instantaneous, but it lasted for about 3 weeks following the end of stimulation. So this is the concept that is basically used in the treatment of depression and obsessive compulsive disorder that is currently approved for RTMS, as of now. And wherein the stimulation is actually inducing neuroplastic effects in the brain, and the mechanisms of which we still are unclear about. So combination of not just RTMS, but combination of TMS and TES also, that is the TACS or TDCS, also seems to be more relevant these days, because we know that in Parkinson's disease patients, there is loss of motor neuroplasticity. That is, they have difficulty in learning, in motor learning specifically. And this has been shown in RTMS studies with, say, a repetitive protocol that causes facilitation or increase in excitability of the motor cortex. So in healthy subjects, which are represented by the black triangles here, you see that there's a clear increase in MAP amplitude after a session of, say, theta burst stimulation, which is basically a repetitive TMS protocol. But then patients with Parkinson's disease, which are the blue and the red markers here, they do not show any increase in motor output. So which means that they have deficient motor plasticity, whether they are on or off medication. But what happened was, in these authors, they combined RTMS, the same protocol, with 70 hertz alternating current stimulation. And they found that the 70 hertz combination with RTMS not only enhanced the facilitatory output in healthy controls, it actually restored the deficient plasticity in Parkinson's disease patients. So you can see the empty squares which are almost similar, which show almost similar neuroplasticity in patients as in healthy controls. So which means when you look at pathophysiologically, you can really say that gamma oscillation, that is the 70 hertz rhythm in the brain, is probably, it is deficient and is probably contributing to the lack in, or the deficit in motor learning or the motor plasticity that we see in patients with Parkinson's disease. So just to summarize what I've just spoke about till now, movement disorders manifest as abnormal movements or posturing, and they cause significant disability. There's no question about that. But their pathophysiology of almost all these disorders remains largely unknown. And that is why we probably are yet to discover an effective treatment strategy for these disorders. Non-invasive brain stimulation in combination with functional neuroimaging methods offer great promise in examining the pathophysiological mechanisms underlying movement disorders. And this is not just restricted to movement disorders. This multimodal approaches that we are working on currently may be useful in understanding the neural basis of other neuropsychiatric disorders as well. So with that overview about non-invasive brain stimulation and movement disorders, I just would want to summarize what kind of work I do in my lab at NBRC, which is the Human Motor Neurophysiology and Neuromodulation Lab. So I have the aim of the lab I have two basic aims. So one is to elucidate the neurophysiological basis of the clinical phenomena that are observed in patients with movement disorders. So there is no, so if you look at the say the pathophysiological features of movement disorders, it is not just one or two, it's not a one-to-one connection. So Parkinson's disease itself presents with a whole spectrum of symptoms, and same with dystonia, and it's the same with tremor. So it's very difficult to pinpoint one symptomatology to one disease.
Nivedita?
Yeah.
I'm really sorry to interrupt you, but we also have a question. So it'd be great if you could.
Yeah, this is the last slide.
Okay, doke. Thank you.
Yeah. So what we do is we try to look at the pathophysiology of movement disorders. Specifically, I'm interested in levodopa-induced dyskinesias, which are basically abnormal movements that occur as a side effect of the standard treatment that Parkinson's patients are given. Secondly, I also look into some physiological phenomena to understand the neural basis of the normal physiological phenomena like surround inhibition and response inhibition, and see how these networks are altered in patients with movement disorders. Another area that I am interested is in motor cognition, specifically talking about the sense of agency, because it is important in patients with functional movement disorders, but that will require a separate talk on its own. I also am interested in developing effective neuromodulatory protocols, which may be employed in the treatment of movement disorders, specifically in optimizing individualized neuromodulatory protocols for patients with movement disorders, and also in developing EEG-informed brain state-dependent closed-loop stimulation. So this is something that has recently come up in the field, and we are trying to develop something like this for our patients with functional movement disorders. So, so thank you to all my collaborators, Dr. Rupa Rajan, who's the movement disorder specialist at AIIMS New Delhi, my colleagues at NBRC, Arpan and Dipanchan, and Dr. Shiv Kumar from CMC Vellore, who is working with me on the closed loop algorithm, and Dr. Schuler from Yale, who will be working with the neuroimaging studies with me. And my funding comes from DBT India Alliance and the IYBA Award and NBRC core funds. So thank you, BiosWatch India, and thank you all for your attention. So any questions?
Nivedita, thank you so much for that really interesting talk. We have a few questions in the question and answer box, but we're running late. So it'd be great if you could actually answer them, type in your answers. And once again, thank you so much. And now Dr. Poonam Thakur will take over the mic for the rest of the day. Hey, Poonam.
All right. Thank you, Anupama. So hello, everyone. Let's continue the momentum we have built so far. Now is the time for the fourth invited speaker of the day. Please extend a warm welcome to Dr. Aditi Bhattacharya. She is currently the lead of operations or publications at CACTUS and is a former scientist E at INSTEM, Bengaluru. During her scientific research, she and her team has worked to identify the circuits that are differentially active when mice with autism spectrum disorders or intellectual disability behave, the proteins that are consistently expressed as a consequences of certain behavior in these circuits, and how the protein changes as brain develops in utero to early postnatal stages. She has served as an editorial board member at Scientific Data, Nature Publishing Group, and also won the 2019 to 21 Bill and Melinda Gates Foundation BIREX Sentinel Grant Award. Today, she is going to represent the neuronal cellular and molecular biology field and will give us insights into investigating single nucleotide variants in neurological conditions such as diagnostic biomarkers to enable drug development and identify potential comorbidity in carriers. So Dr. Aditi, over to you.
Thank you very much, Poonam, and a big hello to everyone here. I am just going to try to share my screen. Please let me know if that is working. But before that, what I wanted to actually say is that, that I'm extremely happy that this NeuroFemIndia has eventuated and actuated into something that's so powerful. I had the opportunity to see some of the ALBA network videos later on offline, and it's so powerful to see that. And I recall having a chat with Shruti very many months ago about this process. And it is very nice to see this, and I hope that this becomes an annual phenomena or even more frequent. So today what I'm going to do is, I hope everyone can see my screen. A quick holler would be nice.
Yes.
Okay, wonderful. Yes. So I am basically just going to today talk about, I would be considered an outlier right now, since most of the other speakers are very well entrenched in academia. I did have an academic journey, and I started my undergrad in DU, and then I did my integrated MS PhD at NCBS, and moved on to doing a postdoc in the US, and then became a research faculty for about 5 years at INSTEM. I have always found that it is useful to be self-aware in amongst all the journey that one does, and doing periodic checks in seeing what really drives you. And what I found was that increasingly, as I was getting more entrenched in looking at the brain, how the brain develops, I realized that my motivations were not so much starting with the wonder and how it actually happens, but more to do with solutions. And this came because of an early exposure to children with autism, with Down syndrome, with epilepsy, as a result of a lot of time spent with my mother, who is a clinical psychologist specializing in childhood disorders. So my induction into neuroscience has been very much dependent on trying to understand why the disease happens, and also because I've been always interested in the patient perspective, to try to find even small incremental changes which can make rather large changes in the quality of the treatment or the quality of life that they have. Now, being solution driven, I realized that in all the rather rich scientific journey which I had, I was missing the way in which I was missing the perspective of how human clinical trials work, the decisions that go into bringing a certain treatment therapy into a wide-scale dissemination for the human population. And that was one of the big guiding factors of motivating me to switch over to CACTUS Life Sciences, where I essentially enable or work with big pharma companies who work in the rare disease space and who work in other spaces in trying to bring out their studies in terms of publications, etc. So I'm learning a lot of skills here. This is very much a work in progress and an experimentation, as most careers are. And in case if we meet in future, I would love to discuss if there is interest on how this particular experiment is running. So one thing that I must say is that I've been exceptionally blessed in having strong indirect female mentors who have, at every stage of my career evolution, given me quite sound, no nonsense advice, sometimes not extremely palatable, but has allowed me to actually take the right decisions and to weigh in on the constant conflicts that a person mostly goes through when navigating work, when navigating how to balance life, when trying to juggle between the roles of being a scientist, a researcher, a mother, a daughter, as well as a wife. So with that, what I'd like to do is that I would like to switch gears very much into talking about neurological disease. Neurological disease in the form of different forms which are there. We just heard an excellent talk about motor functions. We've talked about the evolution of the cortex earlier. We talked about the maternal behavior, which is there. There's, of course, neurodevelopment and all that, all of which has a pretty strong genetic basis. And as with the increasing aggressive genetic screens which are being done, we have a lot of susceptibility, risk genes, etc., and all that. Now, one of the big conundrums that neurological disease faces, and is the reason why there isn't much of new neurocentric products that are coming out from pharma, is the conversion rate. That means that from the large number of preclinical studies which are there, be it in different model systems, the number of drugs which are getting approved in the FDA or the EU, SDA, etc. And that happens to be the lowest for any therapeutic area at a measly 1.8%. Whatever is being done is incremental or is more genetic therapies, biologics, etc., which are to do with the immune side of the story. And a lot of this has been put in because of our interest in looking at model systems and trying to find convergent signatures. Now, in our emphasis in trying to go through all of the genetic, the transcriptomic, the proteomic signatures, and then going to the circuit as well as a network level, and then, of course, going from disease in a dish to an organoid to going from the fly to the zebrafish to the rodent, all of it is in the search of consistent signatures. And what happens in the case of consistent signatures is that, yes, it is extrapolatable. It is something that one can definitely hang one's hat on. But the issue is also one tends to lose out on a lot of information that is coming because of the variation in the system. So then comes the translational disconnect, which happens when a lot of the very, very robust convergent signatures, which have been found in a slew of preclinical models, tends to fail in phase 2, phase 3 clinical trials. And quite often what happens is it leads to frustration from the patient's perspective and at the other end of the spectrum, which is the bedside. And these are actually things that I've heard where my son has a certain kind of a mutation. This was a donor that we were trying to find. You can't find medicines that work. And if the medicines are there, you can't predict whether they will work for this condition. And you expect me to write another check to fund your research. My son responded to this drug. This is more to do with subtypes who tend to respond to a drug and who don't. However, in this particular clinical trial, what had happened is that the global average had moved to a certain case. So the primary outcome had failed, and hence the drug was not actually moved on to the next level. And then, of course, there's sheer frustration that there is nothing there. And something which is important for as an antipsychotic and a medicine for a mood stabilizer is being given to a girl with a certain specific kind of autism. And the mother was actually crying that this is the only one that you have that you can give. So this brings to fore an extremely real world problem, which is that while we are so happy with the advances which have happened in the fields of dermatology, cardiovascular, cancer biology, etc., we still seem to be lagging behind in the case of psychiatric disorders, neurodevelopmental disorders. Rare diseases with enzyme replacement therapies are doing far better. Recently, there was a news of a boy with Gaucher's disease who, because of ERT, which is available now in India, was able to get selected to a state level cricketing team and is able to actually chase his dreams. But this is not actually the case for a large amount of the patients that are there and the families and the caregivers who are also impacted because of this. So why are we at this kind of a scenario? And the reason that we are in this kind of situation is because of the way in which we look at the two things. Basic biology tends to look at the effect of a single gene onto the phenome and then probably then extrapolate it to human biology, but mostly with a firm anchoring to the mechanisms, the main mechanisms which are at play here. However, therapeutics actually looks at it the other way. And both of them are actually stuck at the place of the phenome, which essentially is a sum total of the metabolome, proteome, the epigenome, which is interacting with the environmental effects, with the context and the stimulation which is there. And each of this is giving this kind of a very complex hairball, if you may, of an effect where the key plane effect is quite often being swamped out. The signal is being swamped out by all the noise of the variation that you have there. So there has to be a way in which we have biomarkers that can slice out this noise because of variation in terms of, okay, which one of them actually had a biochemical target engagement? Which one of them showed a change in a EEG pattern or something? Which one of them has got risk mutations in other parts of the signaling network, which may actually affect the outcome? Which of these subgroups hence are going to do better with a certain kind of treatment and which won't? And for that, one has to know the variation which is there, not only in terms of what is the variation within the disease cohort, but also in terms of a healthy healthy biochemical system. Hence, a normative data set is about as much important because it allows one a factor to subtractively identify what the disease thing is. So for instance, and this is something that I thought was rather good, if I want to go from INSTEM to say NIMHANS, which is on the opposite end of Bangalore, I have multiple routes to do so. And Google will actually give me multiple routes. However, when I was trying to do the same thing at the time that we had COVID containment zones, there were only certain specific routes that one could take. And knowing those routes actually increased the travel time from 32 minutes to an hour 40. But it also was very, very specifically constrained at certain places. We had lots of checks. I had to show my permits, etc. So disease is what this is. Disease has a certain way of tweaking it. You will probably get pushback from the biological signaling systems in some way. However, if one does not know what the normative system also does and reads into the variation in that, it's going to be a little hard to also predict what something would do in context of a healthy human being versus that of a diseased human being. So with that, what I'd like to convince you about is that sometimes things that don't look as glamorous to do and may actually consider to be as gap filling may actually be required in order to understand the state better and in order to be able to bring out usable, tangible solutions to help impact the treatment outcomes and the quality of life of that particular condition that one wants to study. I'd like to start or just quickly take you through an example of this, and which was essentially to look at naturally occurring common variants. Common variants are things that one normally in any kind of a disease condition tend to look at as a no, it's just present in the population. It's not something that is of important, is going to cause the actual disease or is going to lead to finding out a target that one can have in order to make it druggable. However, natural occurring variants and the power of the common is something that has to be leveraged or has to be taken into consideration in case if it changes the way in which the treatment can be perceived or received. And this is something that after much process of finding things, we were able to publish last year and is really available now. In case if it isn't, then do please write back to me and I'll be happy to share it. And the work was done by Janani, who was an excellent research trainee that I had, and who also believed in trying to find things incrementally better, has got a lot of satisfaction rather than going and trying to find new mechanisms and new circuits. So a little quick segue into what exactly is autism in case if that is not something that one knows about. Autism is a neurodevelopmental disorder, tends to show up between the ages of 0 to 3 years. And mostly there are changes in certain specific areas of the brain, which would be in terms of social communications, repetitive focused interests, and sensitivities which are there. A lot of the autism is genetic. There are very, very strong variants which have been associated with it. There are chromosomal abnormalities. There are large deletions and insertions which are also known. Now, yes. Protein synthesis being altered is a concept that is known to have a contributing factor to the pathology of ASD, where either there's too little synaptic protein synthesis or too much protein synthesis, which is there. And it comes mostly from evidence from single gene disorders of very high penetrance, which mostly are mutations, which mostly carry a mutation in the translational signaling, or the signaling to the factors that regulate protein synthesis. And those would be like Fragile X syndrome, tuberous sclerosis syndrome, SYNGAP insufficiency, or you can have, or they have been seminal studies in which specific protein translation factors have been deleted, and they also lead to the same kind of autistic-like behaviors in mice. So the signaling pathway that one ends up mostly looking at when talking about control to translation happens to be the MEC-ERK pathway, or the mechanistic target of rapamycin complex 1, or the mTORC1 pathway. And I was interested for a very, very long period of time to talk about S6 kinase 1, which tends to modulate different arms of this particular translation, which is there. Now, S6 kinase 1 about 15 years back was considered to be an extremely happening molecule for the drug translation people. However, it became quickly deemed as undruggable because S6 kinase is important in a slew of other things like cell size, cell survival, transcription, mRNA processing, etc. So while we have inhibitors to S6 kinase, quite a lot of them are not going to go into trials because they will likely be associated with pretty severe side effects in other places. So I believe that right now drug discovery is looking at allosteric modulators, etc. And we'll see if there are more things that come out soon. So this brings me to looking at S6 kinase as a good candidate to look for specific common mutations that would be present in the background of larger mutations in autism. And for that, what we started to do with Ivan Iosifov at Cold Spring Harbor was essentially mine two very, very large but very well curated databases, which were the Simon Simplex collection and the Spark for Autism collection. So in this case, in both cases, there was a lot of controlled data which had been taken. That means that the parents had been sequenced, the child with autism had been sequenced. In the case of Simon Simplex, even a sibling who seemed to be within the normal curve was also sequenced. And a lot of the IQ peripheral measurements were done, and in some cases, the iPSC and fibroblasts were also available. Spark is something that is coming up, but we continuously did an iterative search in Spark, and we also got certain hits. The idea was to be able to find, in case if there were any deleterious mutations in S6 kinase, but if we didn't, which we actually didn't, then what would be the common ones that were there? And did it actually become coincident with any kind of a specific phenotype that were being measured within the autistic proband? And what we found were these two specific mutations which were there in the N-terminal domain of S6 kinase 1. And one of them became extremely interesting, which was the D14N, where every time the D14N was present either in the parent and going to the sibling who didn't have autism, the IQ of the nonverbal, the IQ of the autistic proband seemed to be quite all right. But every time that it went from the parent, inherited from the parent into the proband, the IQ dropped quite significantly. And we found this not only in this particular family, we found it in other 5 families as well. So what happened in these cases is that it probably was coincident, but it became a good candidate to actually check whether or not this kind of a single variant could actually be having a phenomena which would impact kinase function. So the thing that we did was essentially, as we do, we took the S6 kinase construct, the human S6 kinase construct, tagged it with the EGFP, dumped it into HEK293 cells where majority of the signaling first happens. And we wanted to see whether or not the D14N was actually doing something different to the downstream phosphorylation events, and whether or not the same thing was happening with a nearby cousin mutation, which was the E44Q. And lo and behold, what we found is that the D14N, these are the downstream targets, phosphorylation targets of S6 kinase 1, and the D14N was actually causing increased phosphorylation of the downstream targets. The E44Q in one specific case was actually causing a depressed signaling in HEK293 cells. Now, was this translating to protein synthesis, which is what was our first interest? And yes, basal translation seemed to be much higher. Basal protein synthesis without stimulation of anything was much higher in the D14N expressing cells versus the E44Q expressing cells. Now, since the D14N was actually causing something that was quite remarkable in terms of the S6 kinase outcome on its signaling as well as physiology, we also thought, why not look at cell size and cell cycle? And we were not actually able to find too much of a difference in that. And we probably thought that this might be because these are in cycling cells and it may have more of an effect in a differentiated cells, which is something that we came to later. However, somebody asked, and Janani got very interested in it, is that was there a change in the active site or the conformation of the enzyme, which was causing the D14N to be actually very, very overactive? So what we found is that basal S6 kinase phosphorylation itself was pretty active. So is it that it was just constantly in an on position? The problem was that the S6 kinase crystal structure is only known for the kinase domain. And it is not known for these very large, very end areas where this particular mutation was. So we thought that we would take a more functional route to actually answer this question, which is to see if we took a very active site inhibitor of S6 kinase 1, which was actually found for research purposes. And we had this wonderful collaboration with Sentinel Oncology UK to actually do that. Whether or not it would inhibit the mutant S6 kinase 1 versus the wild type. What we found is that just by adding the FS115, which is the active site inhibitor of S6 kinase 1, we found a decrease in the basal translation in the wild type cells. But that made no difference in the cells expressing the D14N. So clearly what was happening is that it seemed to be refractory to even active site inhibition. And the active site probably was also altered, which is a very indirect suggestion to an altered structure of the enzyme. So this is all very well, but then as I said, context matters. So we thought that we would actually test out this particular D14N in series of different cells, which are in different states of differentiation, in different maturity, or even different systems, and see whether it just by being there and being overexpressed, would it actually make a difference? And what we found was essentially that when we took just embryonic stem cells, which is which require fibroblast growth factor signaling into in order to differentiate into a neural lineage, just the presence of D14N in these cells were able to push it more into a neural specific nestin positive kind of lineage versus the ones which were just expressing the wild type S6 kinase 1. This was in the absence of any kind of growth factors which were added. So it was just spontaneously becoming more neuralized per se. If one did it in mature neurons, one again found increased protein synthesis. And in cell cultured neurons, we also looked at We also looked at morphology because S6 kinase is known to affect the arborization of the neurons. We found that the arbors were quite impaired when the D14N was present versus the nice, bushy, robust ones, which are there. Then, of course, one of the good things about being in INSTEM is that one can also ask the question of, well, this is all that's happening in a neural specific lineage. So what happens in the case of something else? And we were able to ask Dhan to try to express these in fetal cardiomyocytes. And what we found was that essentially that even there, S6 kinase is required for cardiac hypertrophy, and the D14N expressing mutants were really, really high in their cardiac hypertrophy markers, which were there. So to summarize, essentially what we had was that we had now a common variant, which is available, which probably is 3 in 10,000. So in a place like India, quite a few of them would actually have this. It's inherited in a dominant fashion and can influence a lot of the normal functioning of that individual, because S6 kinase seems to be pretty central for that. But also, should there be any rapamycin analogs or something being given for any condition that this human being develops, may actually be refractory to that kind of a treatment because of the manner in which S6 kinase is functioning in that context. So these are the kinds of, sorry, I'm going to stop sharing. So these are the kinds of mapping that we need to do so that we are able to give these tangible kind of, usable leads, which would help predict precision medicine in a variety of contexts, just not in the case of a brain condition. So with that, I'd like to end. I hope I haven't taken up too much of your time, and I'm happy to take questions.
Thank you so much, Dr. Aditi, for this profound talk with so many great examples and analogies. I'm sure audience have enjoyed the talk as much as we have. There are a few questions in the Q&A box, but in interest of time, we have to move forward with the next speaker. We request you to kindly answer the questions by typing out there. Let us know if you face any trouble doing that. Thank you.
Thank you so much, Buna.
Thanks a lot, Dr. Aditi. So now it's time for our final invited speaker of the day, Dr. Shantala Hegde. She is an associate professor and consultant neuropsychiatrist at NIMHANS, Bangalore, and she runs a music cognition lab. She and her team is working on both the basic science part and the clinical aspects of how music-based interventions can be used to treat various neurological and psychiatric conditions. She currently holds intermediate fellowship of Wellcome Trust DPT India Alliance. She is an affiliate professional member of Academy of Neurological Music Therapy and has won an award in 2017 Fondaziano Pierfranco e Lucia Mariana Foundation Scholarship. Today, she is representing the cognitive neuroscience field and is going to take us to tell us how music-based interventions can be used to understand brain behavior in cognition, motor function, and emotion to enable therapeutic and clinical advancements. Dr. Shantala, thanks a lot for accepting our invitation, and it's over to you.
Thank you, Poonam. The pleasure is mine, and I'm truly humbled and extremely excited to be part of this wonderful event, so well curated. And I must also thank the previous speakers, especially Nivethida and Aditi, for setting the stage for work related to patients. And because the whole next 30 minutes, I'm going to talk about from a different perspective, more from the clinical aspect. And the last 2 days with such wonderful studies being presented by various eminent scholars and scientists, we have gathered here to understand about the brain and behavior. And I'm sure each one of us are so much interested in asking different questions to understand the functioning of the brain as well as its impact on behavior, be it animal studies or human studies. Being a neuropsychologist and a clinician, I thought let me start my talk with an experience, a case report that we recently published. This was work that we carried out on a 25-year-old young girl who unfortunately met with a road traffic accident. When she came to us, it was already 2 years post her injury. But as we all know now, previous speakers have also suggested that any injury to the brain has its consequences on the behavior. And in a condition like this, definitely depending on the nature of the injury and the severity of the injury, you see consequent neurocognitive deficits, such as deficits in attention, executive function, social, emotional domains of function. What we did with this patient was a holistic rehabilitation. So it was based on the principles of neuropsychological rehabilitation. I'm going to take you through all that in the next 30 minutes. But most importantly, in many of these conditions, this is just an example, but almost all our cases, degenerative conditions or other neuropsychiatric conditions, the question is the sense of loss of self, especially the insight into their deficits and that they are not able to function as they were perhaps just before the injury, and they see a whole different personhood. And this loss and the grief work requires not just addressing neurocognitive functions, but also addressing other domains of functioning of an individual, looking at from multiple domains of functioning of a given individual. So in this case, as expected, we saw a range of neurocognitive deficits. She had amnesia, both retrograde and anterograde, which means she had pockets of memory kind of lost in her autobiographical memory in the past, and she had difficulty forming new memories. There were personality changes. She was irritable, anger outbursts, which had its impact on her relationship with family members and friends. So there were consequent impact on her social domain of functioning, relationships. She couldn't relate to what her friend said who she was, and she said, I'm not the same person anymore. And it led to secondary depression. There were suicidal ideations. So this core thing of loss of self is what we addressed through this holistic work that we did. And this is just one of those cases, but day in and day out being a clinician, 30% of her time goes to clinical work, 30 for academic and teaching and other activities, and 30 for research. So I'm going to take you through the next 30 minutes on neuropsychological rehabilitation that we do in clinicians as in clinical population, and the newer frontiers that is occurring in this field. So as I mentioned, any injury to the brain, be it functional injury or a traumatic injury or an acquired injury, what I mean by functional injuries like neuropsychiatric conditions, where we really do not know where the deficits are, or it's still more to know about what really is happening in the brain, what network is getting affected. Acquired injury would be like stroke or epilepsy or other conditions, or traumatic injury like head injury due to various external trauma that happens. Nevertheless, what happens is a consequence of multi-dimensional sequelae, including neurocognitive deficits. Sometimes pre-existing neuropsychiatric conditions get overlaid on the acquired condition. Person may have had some personality disorder, and post-injury that gets aggravated or magnified. And to address those issues becomes very, very important to help the individual to cope or recuperate from this condition. There could be side effects of the medications itself, like in epilepsy or in children. Medications can lead to neurocognitive deficits, slowness in information processing speed, and its impact on that. Consequently, there are biological functions that get affected, sleep disturbances, that impacts neurocognitive function. So overall, what we're looking at is a complex interplay of all these variables that gets affected, leading to poor quality of life or overall functionality that gets affected in an individual. So what it leads to is basically disability. And what we know now today is that neurological conditions continue to be one of the leading cause of disability worldwide. Over the years, with advancement in medications, pharmacological research and all that, of course, mortality rates have come down by 28% from 1996 to 2016. But what we see is that DALY, that is the disability, years adjusted life year. That means how many years of potential a person could have contributed to the society that gets affected is about 276 million, which is pretty huge. But the bulk of the burden, of course, in neurological disorders and even neuropsychiatric disorders continue to be in low income and middle income countries. There are certain conditions where if treatment is available within that golden period, let's say in head injury or stroke, disability can be withheld to a large extent. As I said, severity of the illness, chronicity of the illness, and duration of illness, all these factors do matter when we are talking about neuropsychological, neurobehavioral sequelae of the individual. The next leading cause of the non-fatal causes of disability or overall burden is, of course, the neuropsychiatric condition. They are the fifth leading cause of DALI. In fact, the leading cause, the first cause are cardiovascular conditions. Even those conditions today we know do have neurocognitive deficit. Oncological conditions do have neurocognitive deficits. So therefore, brain and behavior, this complex interaction and the psychological impact of these conditions is something that needs to be addressed. In India, 1 in 7 people is affected by one or the other neuropsychiatric conditions. And by the recent National Mental Health Survey, we know depression is the leading cause, which actually affects the person's overall functioning. Neurocognitive deficits, understanding of neurocognitive profiling in depression itself is an area which is still emerging and a lot more work needs to be done. But what we know at the core is that irrespective of which neurological condition you're talking about, Nivethida actually dwelled so beautifully on Parkinson's disease, motor functions. In fact, patients, when they have motor deficits and unable to do day-to-day activities, again, they have a lot of secondary mood conditions, depression, or even added neurocognitive deficits because of the known pathways. So when I talk about neurocognitive deficits, what do I mean by this? These are processes, cognitive processes that actually helps us to cope with the environment, to adjust to the environment, to deal with the environment in a meaningful way. So we do need attention, learning and memory, language functions, emotional processing, social cognitive processing, motor functions, executive functions, which all get impacted in these conditions. And why is it important? Because let's say in schizophrenia, which is one of the debilitating neuropsychiatric conditions, functional outcome is determined by the level of neurocognitive deficit. So we do know now that working memory, executive functioning, especially the response inhibition, learning and memory actually determines outcome to large extent, even the relapse rate as well as the functional outcome and the recovery. And what we know is that in schizophrenia, until let's say 3 decades ago, it was kind of intermixed with negative symptoms. Today we know that neurocognitive deficits are central to the syndrome itself, central to the clinical condition. In fact, they predate the onset of the psychotic symptoms and continue to be there in the residual state. So which means addressing neurocognitive functions separately and not like just addressing psychotic symptoms or the negative symptoms, positive or negative symptoms, is not good enough. Let's say traumatic brain injury. As I said, traumatic brain injury, severity of the injury does matter. There's a lot of consequences on the neurocognitive, overall psychological aspects. And even after certain physical injury is kind of addressed, let's say neurosurgeon says, now my job is done. It's up to you to take over and help this patient. So for them, the patient is alive. But for us, how is this patient actually coping in real life, and how is he or she at least functioning near normal to the near normal level? Let's take stroke. What we know is the golden hour, of course, as I mentioned, but post-stroke globally, 1 to 3 months, we see significant cognitive deficits. So prevalence of neurocognitive deficits is almost 50 or little more than 50%. And it can range from mild neurocognitive deficits to very severe condition like dementia. And we do know that patients who have had stroke are also at high risk for degenerative conditions like vascular dementia. Parkinson's disease, it was addressed similarly. They do have certain percentage who go into dementia, but corely at the core of non-motor symptoms is neurocognitive deficits. I'll touch upon that again. So when we talk about, sorry, my screen. Okay, I'm not able to see the top of my screen. There's something. Okay.
We are able to see the screen.
No, I'm able to see one thing. I'm not able. That's absolutely fine. So when I'm talking about neurocognitive deficits and how do we address as neuropsychologists or those interested clinicians who are addressing in rehabilitation with specific to cognition is neuropsychological rehabilitation. In literature, terminologies like cognitive rehabilitation, cognitive remediation, retraining, training have been interchangeably used, but truly neuropsychological rehabilitation is an umbrella term under which many of these evidence-based intervention programs are placed together. What are they? They are basically systematic, functionally oriented service of therapeutic activities that is based on assessment. So assessment is the key. Thorough neuropsychological cognitive profiling of the given individual, psychological profiling of that individual is very much important, based on which you would plan the intervention program. And as I shared in the example, sometimes it ranges from few weeks, sometimes to years when we address these functions and help the patient. It's a wide range of evidence-based intervention, basically focusing on helping the patient to be able to get back those functions that the patient could do before the injury, if not at least be able to compensate for those loss that has happened. So it is to ameliorate cognitive, emotional, psychosocial, and behavioral deficits that is observed because of the injury to the brain or insult to the brain. Cognitive functions, as I mentioned, such as attention, memory, executive functions are specifically addressed. They are tailor-made. And as I said, I cannot stress more on the importance of neuropsychological evaluation. So these are standardized tests administered by clinicians. And we do compare it often with matched healthy control to know how much deficit are we talking about in terms of comparing with age, education, and gender, because we do know cognitive reserve also plays an important role. Sometimes on our test, we may have the patient showing the score that is above a cutoff score, but subjectively the patient will be saying, I'm really bad with my memory. And this is to say that patient has deteriorated, but actually our normative data may not show. So as a clinician, this judgment comes about as to where we are addressing this cognitive deficits. And we also carefully look into the patient's premorbid level of functioning when we are planning and designing for neuropsychological intervention. Just quickly, I cannot go in depth on the history of neuropsychological rehabilitation, but I thought I'd take this opportunity to give how it actually evolved over the years. So it was post-World War, thanks to a lot of World War, a lot of neurocognitive tests, understanding the area of the brain that got injured and its consequences on behavior, cognitive functions were well studied, and then it helped us evolve this field, what we today call as neuropsychology and so on and so forth. But Kurt Goldstein, who contributed by working with German soldiers, but he focused mainly on abstract reasoning back then. The focus was specifically on looking at limited number of cognitive functions or just addressing cognitive deficits like memory issues or things like that. But in the later years, 1980s, work started looking at, we cannot address only the neurocognitive deficits, but also look at the larger picture of psychosocial functioning, what we today call as holistic approach to neuropsychological rehabilitation. And in India, in the early 1970s, 75 is when the neuropsychology lab and the clinic unit started in NIMHANS, and my senior professors, Dr. Shobhini Rao, Dr. Mukundan, all worked on these principles of developing indigenous methods to help our patients specific to Indian population. And I was lucky enough to work and take that work forward for patients with schizophrenia, which I'll touch upon now. But key thing in neuropsychological rehabilitation, there are majorly two approaches. One is a restorative approach and a compensatory approach. Now, restorative approach basically believes in this principle of drill-based exercises. We are talking about helping the areas of the brain that are intact, the networks that are intact, and stimulating those through specific exercises by a sound understanding of the neural underpinnings of each of these cognitive functions as we know today, and help the patient overcome those deficits and be able to acquire the skills that he or she is expected to do. Sometimes it doesn't happen. As I said, the severity of the injury could be so severe, but we could use compensatory approach. But the key thing in any approach that we're talking about is generalizability and ability to transfer this to their day-to-day functioning and sustainability of these improvements beyond the framework of the rehabilitation sessions. Now, the basic rationale behind, I'm sure previous speakers did touch upon, and Shubha Tole also so beautifully mentioned about the developmental brain, and the neural plasticity is the key thing, the mechanism behind any of these principles that we're talking about, be it respiratory function or the compensatory functions. The thing is, you're talking about this dynamic brain, and we are dealing with humans with such complex nature. It's not just one variable we are able to control. But we do know today that these neural circuits can be altered. There is both negative and positive neural plasticity that happens in stroke patients. We see negative neural plasticity. We do address those. But neural plasticity, which is the key veritable nature of the human brain, is the underlying principle behind this recovery, functional recovery of broad array of clinical conditions that we're talking about. Now, evidence people have questioned. Of course, as I said, it's so hard to do. RCTs and meta-analytic studies have been put forth. Initially, work was mainly done in patients with traumatic brain injury or acquired brain injury, but we do have now meta-analysis done in neuropsychiatric condition. Of course, the challenge is the small to large. There's a wide range of effect sizes. Different studies have spoken about. The variations that we observe is due to the methodology, the assessment tools that are used. Now consortiums are being discussed, groups are discussing about the uniformity of assessment itself and how we are measuring outcome. But what we know is that even with single case studies, even Cortex has now accepted that we need to focus on single case reports as well and not really rely only on the RCTs, because when we're talking about human patients, it's very, very difficult to control for myriad variables. But the key thing in neurorehabilitation is definitely to develop modules that can have large generalizability. So briefly about my initial work with patients with schizophrenia, as we know in India, we have patients coming from nook and corner of the country. Not everybody can afford to come to a tertiary center like NIMHANS. So my mentor, Dr. Shobhini Rao, helped me actually in developing both hospital-based and home-based cognitive retraining program for patients with schizophrenia. And in fact, in my hospital-based program, I spent more than 150 hours per patient incorporating family intervention along with neurocognitive rehabilitation. And in home-based remediation, we incorporated a component of psychoeducation to the family members, basically to balance this realistic expectation from family members, changing the family milieu to help patient go back to their near normal functioning with improved cognitive function. And what we found was there was moderate to large effect sizes changes because of the intervention. The home-based program was an RCT. We tried our best, first RCT in schizophrenia and neurorehabilitation from India. We saw changes in attention, executive function, verbal memory, which has an impact on functional recovery. We did see decrease in negative symptoms, which is otherwise very hard to target, and also qualitative changes in their occupational function. Many have gone back to gainful employment, which is what we would want our patients to do. Now, as I said, neuropsychological rehabilitation is a fast emerging field. There are newer methods that are being explored, including virtual reality, computer-based, technology-based intervention, not just the traditional paper pencil based that it began with. The tenets remains the same, the principle remains the same, but different modalities are being explored, and music-based intervention is one of those newer frontiers. And I was frankly lucky to be able to combine my passion, my hobby with my job, my work that I do with patients. So I started off working in the area of music and cognition since just while I was just finishing my PhD, and got the opportunity. I must thank various people and the funding agencies who helped me establish this lab in NIMHANS. So what we know today is that music over the last 3 decades have contributed significantly to our understanding of the brain and behavior itself. Thanks to advancement in technology, like Nivethida said, it's not hard, it's not easy to do imaging studies or EEG studies on humans, and more so with musicians, and looking at the impact of musical training. It's only happening in the last 3.5 decades or so. And music therapy has always been there, but to understand how and why music therapy works, and to give a neuroscientific explanation and to give the reason behind the process behind the therapy is the key thing. And that was one of the lacuna in the field of music therapy. And today we are able to establish the evidence for the impact of how music interventions or how music therapy is benefiting in improving cognitive functions or other domains of functioning. So briefly, what do we know today as we stand in terms of music and from a neuroscience perspective is that neurologically intact individuals are born musicals. We're not talking about only formal musical training. We're talking about this normal distribution of this ability. And every born individual, except the extremes, the ones like in in SLD, there could be some who have amusia, but rest all, we appreciate music, we indulge in music, and music is universal. And music modules, that means different aspects, components of music is, as we know today, is anatomically isolable. That means components of music, I mean pitch, rhythm, timbre, melody, emotion. These aspects are at the neuronal level represented in different areas of the brain, and it works as a network. And it's not, so each of these components are processed differently, but ultimately we perceive music as one in unison. So in that sense, music processing relies more than one module, and they're neurally separable. And the quest is still on to understand more about these components of the brain mechanism and this effect of musical training on impacting neural plasticity. As we stand, we know music engages more than one network, especially the auditory pathways, quite obvious, but also language like music syntax, attention and cognitive, the prefrontal, the basal ganglia, and the cerebellum pathway, memory networks, the motor network, as well as the emotion, the reward mechanism, so enhancing dopamine pathways and working on the neurotransmitters at the neurotransmitter level. So this is a paper we recently published in, kind of putting forth our idea of how neural plasticity is the substratum for music-based intervention in neurorehabilitation. I'll be happy to share the full text in future. Coming to quickly about the work that we are doing, Nivethida has touched upon this, so I'll quickly run through this slide. We are more interested not just in motor symptoms, but also addressing non-motor symptoms, which is quite debilitating for these conditions, especially Parkinson's disease. So neurocognitive functions play a key role. What we know today is when we talk about music and its impact and how we could use it in Parkinson's disease, it's the same basal ganglia, which is also important in beat perception. So we know that beat is one of the important component, or along with melody, when we talk about music, and beat perception engages the very same network, which is also important in motor processing. And thanks to my MPhil students, Vishwas, Babina and Mohit, some of which are still ongoing work, what we have found is patients with Parkinson's disease, when compared to healthy control, that along healthy matched control, that means age, education, and gender, what we found was they have an array of rhythm perception deficit in addition to neurocognitive deficits. And the key thing here is the beat perception, beat discrimination ability, beat perception in musical context. So in a musical context, if you ask them to maintain a rhythm or perceive rhythm, these deficits also predicted their performance in the non-musical domain of cognitive functioning, such as working memory or visual memory, and also emotional processing. So they have deficits in musical emotion processing, also deficits in recognizing facial and prosody emotions through in the musical domain as well. So prediction of deficits in musical domain to the non-musical domain of functioning is quite being established now, which brings us to the point that where we can build evidence to develop new modules to address non-musical cognitive deficits through music-based intervention program. Similar work we are doing with patients with schizophrenia, as I mentioned, neurocognitive deficits, emotional deficits play a central role. What we found in these patients as well in schizophrenia, that deficits in rhythm perception, rhythmic contour, musical incidental memory do have, were highly correlated with musical domain of processing. And facial emotion recognition and multiple domains of musical abilities were strongly correlated. So we're not talking about just mere one connection, but we're talking about music as a rich cognitive process in itself, connecting to the non-musical domain of functioning. And musical emotion recognition actually predicted affective prosody perception in language. So which means that we could help patients to recoup with their emotional deficit, neurocognitive deficits in future through music-based intervention. Quickly, through one of our exploratory study, which Shikha and Rajneesh did in our lab, which was published last year, was looking at one-time music listening in schizophrenia patients. This is chronic schizophrenia patients who are known to have neurocognitive deficits, especially attention, as I mentioned. What we did was an ABAB design. So patients themselves became their control, and we looked at P300. What we did was one group of patients got music listening, followed by silent condition, and then their oddball, the traditional oddball paradigm. The other group got silent condition, followed by music listening condition, and the P300 oddball paradigm. I would be happy to share the details of the study or share the paper with those who are interested to know more. But the key finding of the study was even one-time listening brought out changes in their behavioral performance of accuracy and reaction time. And not just that, we found P300 latency being shorter in patients with when after the music listening condition, and across channel, although it did not reach significant value statistically, but across different channels, we found that the amplitude was far larger in music condition. And alpha power spectrum analysis showed that alpha, post anterior alpha, where patients felt extremely relaxed, was also observed. So it's all these small bricks that we're trying to build for our future work that we plan to carry out in patients with not just Parkinson's disease, which is now currently funded by the India Alliance DBT Welcome Group, and the NIMHANS funding and the UGC and ICMR funding for many of my students. We are carrying out work in patients with depression, children with learning disability. In fact, we recently published our work looking at how rhythm perception is significantly affected in children with learning disability, and how future work can be looked into developing retraining modules based on music rather than language. Children already feel overburdened when you do intervention programs using language again, when it's already threatening for them. So music can be far more engaging. And we do know through Cochrane review and all that adherence to treatment, music-based intervention is far better than the traditional drill-based exercises, which can be sometimes quite boring. So to conclude, the need for neuropsychological rehabilitation is only increasing with growing levels of burden, be it neurological, neuropsychiatric condition. As I mentioned, it's also spreading to cardiovascular conditions, oncology, where psychological burden and consequences are often seen, and neurocognitive deficits seem to be playing a central role. And as I said, assessments, thorough evaluation of patient is extremely important. There is a need for many number of young trained professionals in the field. And it also has to be a multidisciplinary team. As I said, newer techniques and methods require a team from different fields like engineering or as I said, virtual reality, computer-based. We need to develop more methods which are be able to help patients even in remote places. We can't expect everyone to come to a tertiary place, and these sessions can be time consuming. As I said, we may need few weeks to 1 year or 2 years while working with a given patient. Newer methods are emerging, and music-based intervention definitely is taking its lead now, and plethora of work that's happening in the field of neuromusicology, exciting field, and we need more people who are trained in music, clinicians, as well as people from different fields to come together to take this work forward. And there is scope for further research. There's huge need to build a lot more evidence for neurorehabilitation at large, and also for music-based intervention. So with this, I conclude. I thank my PhD scholars who are carrying out work with different clinical populations, my senior research fellows who are being funded by India Alliance Welcome Team, Welcome Trust, and my MPhil scholars who carry out these exploratory work and help me build this short bricks, as I said, for the future work on a larger rehabilitation programs. And really, I must thank India Alliance again for giving so much of support to establish this laboratory. Music cognition is like dream come true for a young researcher like me, and to my mentors, my collaborators at NIMHANS, without whom such work just cannot happen. And once again, thank you all for such patient listening on a Sunday afternoon and the last talk of the day. And thank you, NeuroFem 2021 team, who are so amazing, and I've been enjoying all the talks for the last 2 days. Thank you so much.
Thank you so much, Dr. Shantala, for that amazing talk. I think we do have time to take one question. So, so Arushi Batra asked, what are your views on non-standardized tests that exist today? As much as she's aware that kids under 6, we cannot use these specialized tests. So how are they evaluated in that case? And does it really pose a problem in classifying the kids based on IQ scores?
Yes, there are standardized tests based on age-wise. We do have standardized tests, so we shouldn't be using non-standardized tests at all. We do have normative data. Even for infants, we do have scales. We do have Vineland social maturity scale. So we may not talk about it in terms of IQ, but we may talk about it as social quotient. And definitely, you have to use standardized tests. There's no point just logging in. There are so many tests that are available when you Google search talking about IQ tests for kids, IQ tests for adults, but these are not really standardized. So, yeah. I hope I answered that. Was there any second phrase to it? If I missed it, that's it, right?
I think that pretty much covers the question. I will maybe plug in one of my own. So the music-based interventions that you do. So how do you decide on what type of music, and is that based on what patients, the music they like or they enjoy, or is it something standardized in that sense? And
thank you for that.
For that therapy.
Thank you for asking this question, Poonam, because this is the kind of question I am often asked with. So what happens in cognitive remediation, as I mentioned, you're actually addressing specifically certain neurocognitive functions. So when I said you're approaching music in a neuroscience perspective from a modular approach, you're not just addressing music as, just listening to music. But I'm actually breaking it down into components. So in our program that we are developing for Parkinson's patients, we're actually looking at beat perception, rhythm perception, using some of the principles both from Indian classical music. Let's say you have the Kriya Paddhati, like hand gestures or hand movements, and then mapping it onto the cognitive remediation principles, drill-based exercises, as I mentioned, in terms of restorative approach. So you build these exercises in a hierarchical manner. So it's just not passively listening to music. That's one thing. And second, let's say I'm addressing gait in Parkinson's disease. I use technique like rhythmic auditory stimulation. Here again, I'll be using simple musical cues, but the key thing would be the rhythm and how we would help the patient to entrain to this external cueing to their training of gait. And as I said, you're basically addressing underlying neural pathway, understanding the cognitive function that you're addressing through these exercises. So it's just not listening to music alone.
All right, Dr. Hegde, thanks a lot for accepting our invitation and giving us this insightful talk. I'm sure audience have enjoyed this a lot. And with this, I think it's time for us to put an end to day 3 of NeuroFemIndia 2020. And we still have 2 more days to go, and we'll be hearing from a lot more wonderful neuroscientists who are going to share their unique paths through science and life. And just a reminder for tomorrow's very special event, Ungender Workshop, which is for gender awareness and biosensitization. It's going to happen tomorrow. And in case you have not yet filled the form, which is a short and anonymized form, just so that the workshop people can understand the audience attitudes and suppositions regarding bias they encounter in daily lives. We highly request you to please fill the form if you haven't done so far. And tomorrow, Dr. Kavita Babu, who is an associate professor and intermediate fellow of Wellcome Plus GPT India Alliance at IISc Bangalore, is going to kickstart our scientific session. And she will be giving an overview of neuronal, cellular and molecular biology research in India. So please stay tuned for tomorrow as well. For the engagement in the meantime, you can go to website of Bias Watch India or the Twitter handles of Bias Watch India or NeuroFemIndia 21. And once again, we request audience to consider adding their pronouns to their Zoom names. And with that, let's just close for the day. Thank you, everyone, all the audience, all the speakers for your time and engagement on Sunday. And for all of those who have signed up for the breakout rooms, please go ahead to the Zoom meeting links. You must have received a reminder email at 3:30 p.m. for the breakout rooms. So thank you so much and see you all tomorrow.
Thank you. Bye.